Hello, good afternoon everybody, and welcome back after your lunch. This is the slot when typically everybody goes to sleep, but that is not going to happen because I've got three fantastic speakers for you who are going to keep you on the edge of your seat with their fantastic presentations. So, my name is Alison Grant and I'm the Dean of the Faculty of Infectious and Tropical Diseases at the London School of Hygiene and Tropical Medicine, and it's a really great pleasure and privilege to be here for these celebrations and to have the opportunity to hear about all this fantastic work. Without further ado, I am going to move on, actually a slight bit more ado. Welcome to the people online, and hoping that if you have any questions, we're going to keep all the questions to the end, I think, in view of the fact that we are running quite far behind time. So if you do have questions, please do keep those in mind, and we'll try to take a few of those at the end, and also for anybody who's online, so please do put them in the Q&A. Without further ado, I'd now like to hand over to our first speaker, who is Dr. Efwa Yusuf. Efwa, the floor is yours.
Thank you very much, Alison. I hope you can hear me at the back. It's not time to sleep, like she said. I'll implore you, please, if there's somebody sitting next to you who wants to sleep, just give them a nudge. Let's all do this together. So, yes, like she said, I'm Efwa Yusuf. I'm a senior scientist at the unit, an associate professor with the London School as well. What I want to do this afternoon is to talk about the role of pneumococcal carriage surveillance in pneumococcal disease. So, mostly our focus is disease, but we want to just look at the wealth of work that we have done with regards to pneumococcal carriage and see how that supplements the work that is being done with disease. You have seen this slide in a way, and the focus really has been, we've talked a lot, we know infections are usually high there as a cause of disease, and of course yesterday we talked a lot about the non-communicable diseases, but let me remind you that the lower respiratory tract infections remain a very high cause of deaths in children under 5, and you see also that even among the neonates as well, you do have a lot of respiratory infections. Pneumonia, we were told this morning also, we talked about pneumonia. It accounts for about 15% of all deaths in children less than 5 years old. Streptococcus pneumonia is the most common cause of bacterial pneumonia in children. And pneumococcal disease causes about 300 deaths in children under 5. And we can look at where these children are dying. Most of them are in sub-Saharan Africa. So where does it all start when we talk about pneumococcal disease? Really, the point of entry is in the nasopharynx, and that's where it starts. But interestingly, most pneumococcal colonization, carriage, does not really end in disease. So most children, we've done a lot of studies here that have shown that by 3 months, a lot of people, high percentage, have already carried one of the serotypes that will be causing pneumococcal disease. Though most of them would not cause disease, you do have primary spread to areas around. You can have otitis media, but you can also have it being flown into the bloodstream, and then you would have, my slides are moving before me. Sorry about that, I didn't do anything. Everyone's doing it? Anyway, so yeah, so it does cause local spread, and then you can have bacteremia, and you can have secondary spread, pneumococcal disease, meningitis, sepsis, and pneumonia. But importantly also, colonization really provides a reservoir for spread within community. And so the focus really of my talk would really be that spread and what we have done to look at that in terms of surveillance and see how that also impacts on disease. Vaccines work. We had that this morning, and vaccines do save life. And thanks, Beata, this morning, he really gave us a good historical perspective of all the vaccines that have come up, as well as Ed. So in terms of pneumococcal vaccines, there have been polysaccharide vaccines that started right from the late 70s. We had a 14-valent pneumococcal polysaccharide vaccine, and then we have a 23-valent polysaccharide vaccine. But the interesting thing with the vaccines is beyond that, we also had conjugate vaccines, and these really were game changers. So in 2000, we had the first pneumococcal conjugate vaccine, which was the 7-valent vaccine. And now most countries, with the help of Gavi, are able to implement the 10 and the 13-valent pneumococcal conjugate vaccine. The US have recently started the 15 pneumococcal conjugate vaccine. There's between the polysaccharide vaccine and the conjugate vaccine. One of the main differences is that the polysaccharide vaccines were not used in children. They're not used in children less than 2 years old, and that's where you really have the burden of disease. But importantly also, the polysaccharide vaccine does not have any effect on nasopharyngeal carriage, whereas the conjugate vaccine is able to suppress carriage of vaccine types, serotypes. And this is important because, like I showed you, I mean, for transmission within the communities, if people don't carry it, then they would not now transmit it to other people. So that was kind of a game changer with the introduction of the pneumococcal conjugate vaccines. So why do we need pneumococcal carriage surveys, or what are some of the advantages of having this? First, I'll start with a challenge. Invasive disease surveillance is ideal, and I'm sure Grant is going to talk about that. But there are lots of challenges with diagnostics. We've already been talking about that. There are lots of challenges with diagnostics, and so making the diagnosis in our settings is usually very difficult in terms of resources. There's lots also of antibiotic use, so even when you collect blood cultures, getting a yield from that can be very challenging to set up invasive pneumococcal disease surveillance in many places. With the carriage surveys, they're really quick, cheap, and easy to be done. They give us better understanding of transmission within the communities. Like I said earlier on, carriage can be quite high in most settings, at least in our settings. There's also, while you study it, you're able to tell as you introduce vaccine what really happens to the serotypes within the community. And we have what we term serotype replacement, where as you give the vaccine, even though the vaccines that are in the serotypes that are in the vaccines, you're able to eradicate those, but then you have non-vaccine types coming up within the community. It's also important we're able to use the carriage surveys to predict the impact of the pneumococcal conjugate vaccines as they're being introduced into the population. So you're able to see if there's a direct effect on the children that are being vaccinated, as well as indirect effect among individuals, say their mothers and older people in the community. I also think this perhaps, if we talk about surveillance, especially with nasopharyngeal, it's also an opportunity to, we're now just talking about our COVID pandemic where we're able to collect swabs, so there may be a link where we can link some of these surveillance programs and be able to really make the best out of these. The carriage surveys in themselves definitely have challenges. It's not that it's just easy and quick and cheap to do, so everybody would start doing carriage surveys. They do have challenges. And one of that is the fact that the serotypes differ in their capacity to cause disease. So there are some serotypes that you would just not find in the nasopharynx, whereas they cause a lot of disease. Serotype 1, serotype 5, for example, are such serotypes. The reason for that is that perhaps they spend a very short time in the nasopharynx because they're so invasive and they quickly cause disease. So isolating these ones when you do your carriage survey would be a problem. It's also important to know that sometimes it will be difficult for you to interpret the carriage data in the absence of data from regarding invasive disease. So ideally you would need both. And besides also, if you don't have data that dates before you start introduction of vaccine, you may also have challenges. Like we said yesterday, there's a lot of work being done with genomics and things. It's ideal if you have data already before vaccination and then you continue post-vaccination to be able to understand trends over time. But also there are secular trends. There are other things beyond just introducing vaccine that could be affecting carriage, and that also may pose a challenge in trying to understand the results that we get from our carriage surveys. So with regards to
Recording in progress.
with regards to the work that we do with carriage and then disease, so I've shown you the vaccines that are available, and as you see, the polysaccharide vaccine I said had 23 serotypes, but there are more than 100 pneumococcal serotypes. Yes, we've done studies to show that there are only few specific serotypes, and that also includes even the pneumococcal conjugate vaccines. So what we have seen post-introduction of the pneumococcal conjugate vaccines, our target is to reduce disease, so it has done that. So there's been a remarkable reduction in disease in most settings, even in our setting as well. But I mentioned about serotype replacements, so slowly you see that you have non-vaccine type serotypes that are appearing in disease, not so much. It's mostly that, yes, we have. So overall, pneumococcal disease has decreased. Can this stop changing yet? So overall, you have a decrease in the burden of pneumococcal disease. It's the same also with carriage, in that now that we have introduced the pneumococcal conjugate vaccine, we've seen a decrease in the prevalence of vaccine type serotypes that are being carried within communities, but we have also seen serotype replacement where there is non-vaccine type serotypes increasing. And for carriage, in most settings, what that means, it seems like overall carriage remains the same. So you've decreased vaccine type, but you've increased non-vaccine type. There are a lot of studies that we have done here in the Gambia, and a lot of people have contributed to this work using several carriage studies. The first ones were way back in the 90s, where we tried to just understand transmission within communities, and there we were able to see that children are really the main drivers of pneumococcal carriage within communities. There have been several conjugate vaccine trials that have been conducted here in the unit, and a lot of these trials as well. And this morning we had about the PCV9 trial, which was one of the trials that WHO used for licensure of the PCV7 as well. And then there have been a lot of impact studies that are being done here. There have been studies also looking at the microbiota and just looking also at environmental changes as we talk about carriage. And there's an ongoing study now looking at pneumococcal vaccine schedules, and that also has a carriage component to it. These studies have been across the country, mostly in the upper river region where we have the Basse Health and Demographic Surveillance System. That's where some of the studies started. The first study I talked about, where they understood transmission within the community. And for that one, they were looking at children who were sick, febrile children coming to hospital, as well as healthy controls, and the household contacts as well. And through that, we were able to determine that children were the drivers. And the first time serotype replacement was recorded was here in the Gambia, also in Basse, following a vaccine trial that had been done using the PCV5, where we found out that, yes, vaccine type serotypes decreased, but there was an increase in non-vaccine type serotypes among the children that were in that study. We've also done quite a number of studies also in the peri-urban area. We've done a lot of studies in Sibanor, and I would talk about a few of those as we go on. But through these, we've been able to understand accusation of carriage. We've been able to understand duration of carriage. I mentioned earlier that some serotypes don't stay long. We carry them very shortly. They're very invasive, and quickly they're able to cause disease. So we've been able to contribute data to that. We've been able to look at hide effect. So yes, we are directly vaccinating the children in the community, but we're seeing an impact in older people in the community as well. And there have been studies also looking at the microbiome as well. But there are other studies that have been done at the Bundung Maternity and Child Health Hospital, and I'll also allude to that as well. So for the Sibanor vaccine trial, the Sibanor studies were started in 2006 thereabout by Philip Hill, and then Professor well selected within the area of Sibanor. These studies were done within the context where we had a cluster randomized trial that was using pneumococcal conjugate vaccines 7. And then we also had, so following the trial, this is one time point, half of the community were giving PCV7 and the other half, the villages were divided into two, 10 and 11. We also have the national EPI introducing the PCV7 in 2009 and then switching to PCV13 in 2011. So we had a series of cross-sectional studies that were done within this community. And setting is that 1 to 2 years children reduced vaccine type pneumococcal carriage across all age groups, which was kind of indicating a hard effect in the non-vaccinated older children and adults. By this time, which was 1 to 2 years, we did not really have any significant serotype replacement. And then 4 years later on, when the fourth cross-sectional survey was done, we saw that the decrease in vaccine type serotypes was sustained over time, but again, there was really no serotype replacement. So there was decrease in vaccine type as well as from the cluster trial, we had PCV7 from national immunization, and then we had PCV13. So 5 years after that, we went back and we confirmed pre-vaccine status. Seems like they had plateaued, and they were really at a high level. It's around 10% thereabout. And then the non-vaccine serotypes had increased in all age groups. We did another set of cross-sectional surveys at Bundung Maternity and Child Health Hospital. Here we were looking at infants who were 6 to 12 months old, and they were given 3 doses of PCV13. We also swabbed their mothers as well. What we found out here is that we saw the same thing where vaccine type serotypes decreased, non-vaccine type serotypes increased, both in the children, the infants, as well as their mothers. There was a little bit of increase in the overall carriage for their mothers. Okay. We went further to look at some genomic analysis, and this is another way in which some of the carriage studies can be used. Basically what we did, the non-vaccine type serotypes, they were non-typables, so these were we weren't able to type them as pneumococcus per se. So these were sent to Sangha for further analysis. And what we found out was that these were mostly vaccine type serotypes that had lost their capsule. We did this whole genome sequencing with further non-typable serotypes from other studies, and the story was still the same, in that they were mostly non-typable as isolates that had lost their ability to express a capsule. So I just want to put this in perspective to what is happening in other settings as well. So what we found out is that, yes, we have seen a decrease in non-vaccine type serotypes, but this is definitely different from what you're seeing in high-income countries. And some of the reasons for that may be that we had high prevalence even before vaccine was introduced. The vaccines that are being used are different. Some countries are using 13-valent, some are using 10-valent. The schedules are different. If you look here, South Africa, the vaccine type post-vaccine seems very low. And also, some countries introduced catch-up, some did not, some had boosters, some did not. So these are some of the different reasons why you may have these differences. But one message is clear, is that vaccine type serotypes post-vaccination seem to be much higher in low-income countries compared to high-income countries. So in conclusion, there has been an observed decline in vaccine type pneumococcal prevalence in all age groups. We've seen that. However, the vaccine type pneumococci continue to circulate within our communities. The overall carriage does remain unchanged, though we saw a net increase among older individuals in one study, so there is more room for us to continue to understand what is happening. We found out that non-typable pneumococci increased in all our post-surveys. This is at the coast here and also at Sibanor, but this is a little different from what was found in Basse. In Basse also, we've done 3 different surveys, and those ones, vaccine type serotypes within households, it would help us to understand replacement in disease, because ultimately, if you have it in carriage, you want to really understand what is happening in disease. And there may be a need for us to consider booster doses and really consider our vaccine schedules. So there are lots of people to thank here. I realized that even in trying to group people, I did miss the lab. So thank you very much.
So thank you. Can you hear me with this? Yeah. Thank you so much, Efwa. That was a really fantastic tour de force, an amazing body of work. Thank you very much. And even in a reduced period of time, even more impressive. So thank you very much indeed. We're now going to hear more about pneumococci from Dr. Grant Mackenzie, also from MRC the Gambia. And just in case you were wondering, no relation. If you do have questions for Efwa, please do keep a note of them, and hopefully we'll have time for questions afterwards.
So thank you very much. I'm going to race, so you really need to concentrate if you're going to keep up. I've been very much thankful for the other speakers today. Beata and Ed and Efwa have really set the scene very nicely for this study. I really want to tell a bit of a story about how pneumococcal disease control has developed over time and how surveillance, trials and other aspects of interacting with policy and decision making are important in the objective of trying to control disease. So we know that pneumococcal disease is quite common. It's a big problem globally. This study was published before the introduction of pneumococcal vaccine, showing that around about 800,000 deaths per year were estimated to be caused by pneumococcus, and around about 11% of all global deaths in children by pneumococcus. Efwa mentioned that this is now down to around 300,000 deaths per year after reasonably widespread introduction of vaccination. Other speakers have alluded to this particular trial, which was conducted in Basse and Bansang in the early 2000s, where a 9-valent vaccine was introduced in a trial showing good efficacy against vaccine type 77%. But importantly, you'll see the asterisk against in red showing mortality had a significant reduction in the vaccinated group of 16%. So this is a 9-valent vaccine with imperfect efficacy. This really indicates that pneumococcus is causing more than 20% of all child mortality in this area. This trial led to WHO recommendation, but we still had to ensure that there was confidence for routine use in low-income countries, whether it could be scaled up globally, and the costs involved. Our research has been focused on disease in the rural area of the Gambia. You can see in the eastern part of the Gambia in yellow is the Basse Health and Demographic Surveillance System. In pink, you can see the Fuladu West area, that's around Basse and Bansang. These allow us to conduct surveillance with population denominators and more easily follow-up of the population. It's quite a challenging place to work. You're very welcome to visit the field station if you're feeling up to the challenge. The project that came after the introduction or during the introduction of the vaccine in the country was called the Pneumococcal Surveillance Project, and we conducted surveillance in all ages for invasive pneumococcal disease and radiological pneumonia for 10 years. The 7-valent vaccine was introduced in 2009, the 13-valent vaccine in 2011, and we had very clear standardisation at the different steps of surveillance. That was referral, diagnosis and investigation, working very closely with our Ministry of Health colleagues in all Ministry of Health facilities in the study area, where our staff would work with the Ministry of Health staff, providing attention to any child who comes. So I really like this work because it's very practical, because we're actually providing care to any sick child. This was any sick individual who comes to the health facilities. The analysis was limited to cases who are resident in the study area. We were looking at incidents before and after and trying to adjust for potential temporal changes over time with adjustment for the presentation rate of patients so that we can have an interpretation that assumes the presentation rate is constant. Just a picture of Basse. It's a fairly hot, dusty trading town. Again, if you're up to the challenge, you can enjoy the warm weather that we sometimes have. Sometimes you need to be very careful where you put your feet as well. Other dangers may be lurking. And we've had a number of floods where we've had to evacuate and boat ourselves out. But we've got a new bridge now, so we shouldn't have to worry about this now. This is a summary of the surveillance study findings. You'll see at the top of this table, the younger children. We found after 10 years of surveillance, comparing the first 2 years to the last 2 years, an 80% reduction in all invasive pneumococcal disease, a 92% reduction in vaccine type invasive disease. Non-vaccine type disease, as EFWA alluded to, or otherwise known as serotype replacement, was not a significant issue. We used non-pneumococcal bacteremia to indicate whether there may have been changes in the population in terms of risk of bacterial invasive disease in general. We found no significant change in the incidence of non-pneumococcal bacteremia over time, which increases our confidence that the changes that we're seeing is due to the vaccine rather than other general changes in the population. Older children also had reductions in disease, but we had small numbers of cases. If we looked at pneumonia endpoints in the younger children, we had 33% reduction in radiological pneumonia, reduced hospitalised cases of pneumonia, and a somewhat surprising 60% reduction in really severe hypoxic pneumonia, and which is the type of pneumonia that causes mortality. Older children also had reductions, but lower incidence. So the impact of these studies has been published in different locations, really establishing the effectiveness in low-income countries. There was multiple contributions to policy along the way. We also looked at cost effectiveness because the Gavi international funding program for subsidised vaccine in low-income countries does stipulate a 20 cents per dose co-payment for the lowest income countries. That increases as a country increases its gross national income per capita over time. So it's important to consider cost effectiveness. And the real important question here was, if low-income countries transition away from Gavi support and they have to pay the base cost of $2.90 per dose, will they be able to handle that budget impact? So working with EFWA, we determined the cost of introducing the vaccine, the economic burden of the disease, and the cost effectiveness. We compared in some degree to other low-income countries, where PCV had been found cost-effective but often not cost-saving. It's important to note that these other countries had a gross national income above 1,000 per head per year. Gambia's gross national income per head is around 700 at the moment, and there are different Gavi policies for these types of countries, and it determines how quickly you have to start paying additional co-payment. The Gambian program was cost-saving until the country had to pay 66 cents a dose, and even at the Gavi price, the base price of $2.90, the program remained cost-effective. So this gives more confidence to countries on what they really need to do to sustain the program. But there is a problem for pneumococcal disease control, and that is access globally. You can see here a summary of countries where there is reduced access because of poor coverage or countries having not introduced the vaccine at all. And around about 50 million children cannot access pneumococcal vaccine. A number of countries haven't introduced it at all. So the cost really limits global coverage and global impact. And so our most recent question is whether, instead of having a 3-dose schedule, which is the usual schedule in most countries, can you have a 2-dose schedule and save a third of the cost? And from our cost effectiveness analyses, we know that a country costing of an immunisation program is by far driven by the vaccine cost. So the global cost that Gavi has to provide per year is about $1 billion, just for pneumococcal vaccine. In terms of national costs, I've mentioned that countries have to pay around about 20 cents per dose. After transition, it's $2.90 per dose. Middle income countries have to pay on the open market. It's even more difficult. And just as an example, the country of Ghana, before transition, paid about $200,000 each year. But after transition, it's going to be $3 million. So that's a challenge for the Minister of Health and the Minister of Finance to see eye to eye on why they should be, increasing the expenditure by 10 times. So there's also an issue of injections in immunisation schedules. You can see the Gambian schedule here. It might be that we have injectable polio vaccine, and we have reduced doses of oral polio vaccine. So we might get some more IPV if polio eradication goes ahead. We've got a meningococcal A vaccine now, maybe typhoid, HPV now. And so there is an issue with increasing injections. So again, back to our pneumococcal vaccine schedules trial. I'll just tell you a little bit about that. It's a collaboration between the Ministry of Health and MRC. It's a non-inferiority cluster randomised trial in Bassay and Fuladoo West, using a 1+1 schedule where you give an early dose at 6 weeks and a booster dose at 9 months, or the standard 3 doses early in infancy. We're aiming to enrol 40,000 children over 4 years. We're currently at 27,000 with endpoints of carriage disease, immunogenicity and acquisition. And this is just a schema of the trial showing at the beginning or at the top in brown how the clusters are assigned to the 2 different schedules. The effect of those schedules develops in blue over time in the different schedules. We do surveillance for immunisation status in individual children. Our secondary endpoints are really safety endpoints of disease. Our primary endpoint is vaccine type carriage prevalence in children with pneumonia. We've got secondary endpoints in unimmunised infants, carriage surveys in all of the population. We'll also be doing economic studies, mathematical modelling also of how the effect may change with varying coverage of the booster dose, and also decision-making surveys in several countries to understand the decision-making process if a country was to consider this. So we want to inform WHO policy with evidence from a typical African setting with disease endpoints, and also modelling the effect of the booster dose as its coverage may vary. There's studies also in South Africa, UK, India and Vietnam that will be combined together into a body of evidence. And our assertion is that if this herd protection dependent strategy is non-inferior in our high transmission setting, the results should be generalisable to other settings with lower transmission. So results so far of this 1+1 schedule in different locations in the UK, they compared this schedule to their 2+1 schedule, and really showed that immunogenicity is non-inferior after the booster dose for vaccine types. That actually led to the UK national implementation of the 1+1 schedule. So it's the only country in the world, the UK, who is using this schedule at the moment. South Africa also showed non-inferior immune response of the 1+1 schedule. Vietnam has shown non-inferiority in carriage, but we don't have any African data on 1+1 compared to the 3+0 schedules. No data on the booster effect, on actual observing acquisition and the duration of the effect. No disease endpoints, and as I've mentioned, the decision-making framework for countries to use and booster dose coverage. So we're going to try and provide this evidence and facilitate modelling and decision tools. But there is a threat of vaccine type transmission that EFWA alluded to. We're still seeing around about 15% prevalence of vaccine types in a number of African countries, quite different to affluent countries where it's like 1%. Whether this will actually cause breakthrough disease with vaccine types in a 1+1 schedule is unclear. We'll be able to give some information on that question. You can see in this slide on the top left, after introduction in 2009, the dark blue line has fallen with the vaccine types falling to around 15 or 17% prevalence, but still around about 15% prevalence in older children, but also in adults, significant prevalence of vaccine types. There's also a threat of serotype replacement. I did mention before that we hadn't seen this as much of an issue in disease, but I'm just showing you here some data from our disease surveillance up until 2021, and you can see on the left-hand column that non-vaccine type invasive disease cases per year in the Bassay DSS area often vary quite in the absolute number. In 2014 we had 21 cases, and then it went down very low in 2017, and we were all very happy. But in the last 3 years it's been 7, 8 and 18 cases, so we are concerned that this serotype replacement may be developing further as a phenomenon. Reassuringly though, in the second column, the vaccine type cases continue to be very infrequent, with only 2 cases in the last 3 years. So the prospects for disease control and unanswered questions for us to consider. This 1+1 schedule, there is still a lot of considerations before a global recommendation would be made. Particularly a decision-making framework for WHO and countries needs to be established. It's quite a complex decision. We need to have more confidence about whether vaccine type disease would be controlled, whether herd effect will be significant, and whether it will reduce costs enough to actually change decisions. Ed mentioned the 10-valent vaccine from Serum Institute of India, which has recently been licensed. It's $2 a dose compared to $2.90, which is substantial, but the question is whether serotype replacement may be limiting its impact. Is there serotype replacement in low-income countries? This is a real bugbear of mine, because there's about 4 places in Africa where you can do reasonably good quality surveillance. And so we don't know whether there's serotype replacement in many African countries. The higher valency vaccines need to have their efficacy and cost established. Whether residual transmission and disease control will be linked is an issue, and then funding for surveillance in low-income countries is incredibly difficult to secure because no one really wants to fund surveillance. It's very expensive. And I'm going to acknowledge many people, many institutions and funders for this work. Thank you.
Thank you very much indeed, Grant, and thank you for helping us to keep very tightly to time. Again, you'll covering a great body of work there and really important stuff. So thank you very much indeed.
Sorry, Alison, we have a problem. Alfred sent his very good, and it got uploaded so big, so they've now gone to MRC to upload it. So we now suggest they have question and answers,
Okay.
and Alfred present late after the short presentation.
Okay, no, that's fine. Okay, so Alfred's talk was a bit big and isn't yet uploaded, so we don't have it uploaded yet. So what we're going to do is take a few questions at this point, and then we'll hope to come to your talk very soon. So, do we have, let me just see if we've got any questions online. Let's see. No, it sounds like we don't. Okay, you can just wait for a second. Okay, so we can go to any questions from the floor. So, do we have any questions for the first two speakers? Because if not, while you're... Oh, we do, at the back there. Yes, gentleman with the raised hand at the back there. Do we have a roving mic at all? If we don't have a roving mic, then maybe you can just shout and I'm going to repeat your question. Or is that, can you come to a mic? We have a roving mic is arriving, thank you.
Thank you very much, Afua and Grant, for very nice presentations. So I have two questions, and they both go to Afua. So the first question is, in one of your slides, you've shown a table wherein there was an increase in the carriage of non-vaccine serotypes, as well as disease due to non-vaccine serotypes. Well, it seems like the rate in the increase due to disease was smaller than the carriage. Is there any explanation for this? And then the second question has to do with another table that you showed, wherein the decrease in the change in vaccine serotype in South Africa was similar. The rate was similar to those seen in other developed countries, like in the US and UK, when compared to other African countries. So I don't know why this disparity is. Where in South Africa is in Africa, but its rates were much more similar to other countries outside of the continent. Thank you.
Thank you very much for those questions. So with the first one, when I showed that little table, that was kind of deliberate, because what I was trying to illustrate is the fact that in carriage, we have seen replacement, where we have a lot of non-vaccine type serotypes circulating. But in disease, it's not necessarily the same. And I think Grant's slides also showed that already, that in Basse, he showed you the numbers, that vaccine type serotypes, though they seem to have increased in disease, but not so much. So the point is we need to continue surveillance to really understand whether these serotypes that we're seeing in carriage, do they still have the same ability to cause disease? That's basically it. So what we're seeing at the moment is that the serotypes that are circulating in the community, the non-vaccine type serotypes, they are not necessarily causing disease at the same rate as what we had before vaccines were introduced. I hope that explains it. And with the second question that had to do with...
It was about South Africa.
South Africa, yes. So those are some of the differences. And like I pointed out, South Africa so we are a low-income country. South Africa is a little different. One of the differences that is marked is the schedule that they use. They're using 2+1, and all the other countries, most of them is 3+0. So that's one difference already. They have a booster dose, and we don't have a booster dose, so that's one difference. So those are some of the differences that we are trying to tease out. But like I mentioned, the pre-vaccine prevalence for most of our settings was much higher than the others. So there's still a lot of questions. And that's why some of the studies like what Grant is doing is important. Yeah, thank you.
Thank you. Thank you. Any more questions? Any more questions from the floor? Perhaps I can ask one following on from that previous question. I'm risking displaying my ignorance of pneumococcal here, but I was wondering something similar. So is there any suggestion that the non-vaccine serotypes are becoming more virulent? So I mean, to some extent they do have some virulence clearly and do cause disease, but is there any suggestion that they are increasing in virulence? I'm just wondering whether they might be sort of moving into a niche that's been emptied by, that's been made available by the reduction in the vaccine serotypes. That might be a question for either of you. It could be a very bad question.
Well, it's a question. So, for what I know so far, the invasiveness, the ability of the serotypes to cause disease has not necessarily changed. So yes, it's creating a niche. So 1 or 2 serotypes have been able to cause disease post-vaccine, like serotype 12F is non-vaccine. So that's what is being used now to develop some of the other vaccines, like when you have PCV 15 and 20, those are some of the serotypes that are being included. So I don't know if you want to add anything to it, but the invasiveness really has not changed post-introduction. But of course, when you create, if you remove something and you're creating a niche, then there's some interference. So.
I think it's not clear, but it's probably that the invasiveness has not really changed, and that the reason why you might have serotype replacement is that you just have a higher rate of acquisition of those non-vaccine types, and usually instead of seeing as those types once per year, if there's no vaccine types around, you'll see it 10 times a year, and even though it's not so invasive, you might get a little bit more disease.
Okay. Any more questions from the floor? Yes, one over there from Ed, I think. Thank you.
Thank you. This is really a question for Grant, I think. How, in terms of your cluster design, how confident are you that you won't have a lot of contamination between clusters, which I think ultimately would end up, you might demonstrate non-inferiority because of spillover between clusters and contamination between clusters. So is there a way you're sort of assessing movement around between clusters so that would cause contamination essentially between the different clusters, I suppose?
Yeah, thanks for that question, Ed. When we designed the clusters, we had all the schools in the study area mapped. And the attendance mapped as well. So we tried to make, because the most important mixing across clusters might be probably with school attendance of children. So our design of the clusters did take that into account to make sure that those who are resident in a geographical area were going to schools who were in that geographical area. In terms of actually monitoring whether there is interaction, the only way that we can do that is using the DSS in terms of migration, but that's really not getting at the issue of mixing from day to day. I think mixing or contamination across the clusters should be limited, mainly because the intensity of transmission of pneumococcus, you would imagine, is highly related to the household where people live. People sometimes go to the market in another village or basse, but there's not a lot of constant movement around the different clusters.
Thank you. I could ask another question, actually. following on from that a bit, if there's if nobody else is asking one, and how long are you? How long? At what point will your endpoint be assessed? Just in terms of moving, because obviously everybody was on a 2+0, 3+0 schedule, and now obviously you've gone to a 1+1 in some of the clusters. Obviously that's the comparison. How long after you're in the 1+1 schedule will you assess the primary endpoint?
Yeah, so the endpoint is at the population level, and it'll be measured in the 4th year. So we'll look at all children who come to health facilities with clinical pneumonia in the 4th year. So that cluster of population will have had at least 3 years of development of the herd effect within a cluster before we measure the primary endpoint. I'm not sure whether that answers the question.
Yeah, that's fine. Thank you.
Great. Fantastic. So we've now resolved our technical issues, and we're going to shift gear and start talking about malaria. So it's a great pleasure to introduce Dr. Alfred Ngwa, also from the MRC unit here at the Gambia, to tell us about pathogen genomics. Thank you.
Yeah, thanks very much, Alison. I see Mbota smiling because I think this has been a recurrent problem. Every time we've had to do a presentation, my slides don't load up on time. Beate also noted that as well. Anyway, it's a pleasure to be here, Alison, and thanks very much for inviting me to do this presentation. And I will just try as quick as possible to just demonstrate how we've used genomics, in this case for malaria, and try to focus a bit more on anti-malaria resistance and not malaria resistance as it's in the brochure. Because malaria resistance is broader, and it could actually include resistance of humans to the pathogen, where genomics can be applied as well to study it. Malaria, of course, is still a very important disease, and it's been in the headlines a lot lately because of description of its decline in our populations. And as Beate alluded, when you go to the communities and you're talking about vaccines, they ask, what about malaria? And this is evidence of what malaria has been ravaging, the ravage it is causing in our communities. And our mothers are sensitive to this. Unfortunately, that decline has stalled. And in the last couple of years, there seems like there has been an increase in the incidence of malaria in our communities. And following COVID, there were about 200 more cases, 200-something thousand more cases of deaths added due to malaria across the globe. More than 95% of these is in Africa. And so it's still a problem with us and that we need to resolve significantly. A lot of the reasons why malaria has stayed with us has been because the interventions have had problems, and there have been several interventions. When RDTs came, the rapid diagnostic test came, it was a revolution in testing and treatment of malaria because you could rapidly test for cases and then you treat them with drugs. And this was great, but unfortunately, there was development of resistance. There's development of resistance to RDTs, where some parasites in some populations have deleted the antigens, the genes that code for the antigens that are used for testing. And so there is need to survey for this. And the WHO is also posing that we should use genomics tools to actually survey for this. The second case, of course, is treatments. There have been several malaria drugs developed over all the years. Multiple drugs have been developed, and it has faced similar issues, which is what we're going to dwell on, the issue of resistance. And lastly, there are different methods that are used for prevention. And some of that actually includes drugs and the use of combinations of drugs that we already know as single and as monotherapy, there is resistance to these drugs. There is also prevention from mosquito bites using insecticides and bed nets, but mosquitoes have equally developed resistance to the insecticides. And so molecular surveillance for resistance to insecticides is an important component of helping to drive the agenda for malaria elimination across the continent. And lastly, of course, there is vaccines that are being introduced. And there is the RTSS, which is not really highly efficacious. Umberto has worked on that before. And there are evidence that it's selecting actually for non-vaccine types in some populations. And this evidence comes from genomic studies. There is the R21 that has just come in the last couple of years with trials that are going to go on in the Gambia. And we need to use the tools to check our population that these new vaccines are not selecting for non-vaccine types. Now, going back to drugs, I'm just showing some drugs that have been developed over time against various stages of the parasites. And all these monotherapies that you're seeing on the right side, on the life cycle of the parasite, all of them, resistance has developed, especially in the most deadly form of the parasite, Plasmodium falciparum. Now, resistance to the oldest drug, quinine, was recorded as far back as 1940s. So it's not something new. And it's something that the parasite over time has developed its sensitivity to. It's developed the ability to be able to sense drugs, select for the most fit parasites in communities. And our job now is to look for what markers are available that can be used to track this development of fitness against the drug and tolerance. And what we are doing now, obviously, is to help the NMCPs to optimize their drug combinations by surveying for these markers. Now, if you go to the Worldwide Anti-Malaria Resistance Network, they publish this, they have this mutation surveyor. So you can go there and actually look into the map of the Gambia or across the globe for various markers. And what I'm showing you here are markers for all drugs that have been used. Chloroquine, SP, sulfadoxine pyrimethamine, and now the artemisinin derivatives that are used in combination with long-lasting partners. So you can see that for all these drugs, they have been resistant development. Now, it's displayed here by the markers. Now, in the last case here, for the LSHTM, that is responsible for resistance against artemisinin, the markers were not largely seen in Africa in one of the studies that we did, but recently they are actually emerging, and that actually emphasizes the need for us to do more studies. Now, countries have changed their drugs. They're now using artemisinin combination drugs. Different countries introduced these drugs at different times. They've been used for over 20 years now across Africa. And 20 years is a long time for the parasite to develop resistance because it took 5 years to get the resistance strain in the lab against the artemisinins. So 20 years is a long time. Obviously, in the lab is different because the factors in the humans that actually influence the way parasites grow and transmit do differ, and we can't find those factors in our labs. Now, there is a problem because, to me, it's a problem, but in some cases it's good because we have different combinations of drugs used by different countries. But it creates a dynamic because populations don't have these boundaries. The parasites don't know these boundaries. They go across countries, across communities, and they experience different drugs at different times. And that also creates a different dynamic. So they respond to one drug in one population, then there is a migrant with a parasite to another population, and then the parasite faces another drug, increasing the pressure on the parasite to develop resistance. And that's the case in Africa, obviously. Now in Rwanda, in Uganda, and in Tanzania, the markers for resistance to artemisinins are actually coming up. And we are looking forward to surveying these markers. Now, at the continental scale, for us to get this survey right, we need to come into networks. And there is one of the networks called the Plasmodium Diversity Network. It has now been converted into the Pathogen Diversity Network Africa, and its principal mandate is to use genomic surveillance as a tool to track parasites and track evolution of parasite against interventions such as drugs and vaccines. And they're working very closely with the Malaria Gene, which is a program at the Wellcome Trust Sangha Center that provides the platform for sequencing pathogens. And we've sequenced thousands of parasites across the continent using the Malaria Gene. Now, this is just to show you what this kind of consortium can allow us to do. Now, the first thing we've tried to do with our data that we generated with the Malaria Gene is to understand whether there are subpopulations, whether parasites in different parts of the continent form small groupings that are similar to each other more than those from other regions. And here, using markers, we've done this with SNPs, which are single nucleotide polymorphisms. But we are now repeating these analyses with microsatellites generated from whole genome sequencing data collected with the Sangha, with collaboration with the Sangha, across 20 countries. And this is defining not only the substructure of the populations in Africa, but almost kind of alluding to the history of parasites and the origin of the parasites, the time that it might have jumped and where it might have jumped from humans, from non-human primates into primates. And I'm now suspecting, I don't know, Dave and I can have a conversation about this later. It now seems that the parasite may not have jumped into humans in Western Cameroon. It might have been somewhere around the Congos, and this might have been from gorillas to humans. Now, with the genome sequencing data as well, this is agglomerated data. We can look at how related, which parts of the genome are the parasites most related. Now, strikingly, what you see is that regions in chromosomes that code for drug-resistant markers are the regions that are most related, in which case parasites are sharing similar haplotypes around those regions that are allowing them to be resistant to drugs across different populations. So drug resistance spreads with populations and by sharing those regions of the genome. Irrespective of recombination in West Africa by high levels of transmission, we still see these strong markers. And some of the new markers we are seeing there on chromosome 10 and on chromosome 12, for example, the one on chromosome 12, for example, has been further exploited by collaborators in London, Colin and his students and trainees have explored those markers in that region and clearly showed that they increase the resistance of the parasites to the currently used drugs. So it is important for us to obviously to continue to survey these markers. Now, one other thing I did in this and with this data is to ask the question whether the background of these markers we are seeing are the same across countries. In these analyses, we are seeing that the background of these markers form 3 clusters. There is one cluster that everyone shares, every population, West, Central, Eastern Africa, they share. But there are very unique clusters of markers that exist in certain regions, like in West Africa and in Central Africa. It is indicative that, irrespective of the fact that there are common markers of drug resistance that may have been due to spread from East Asia into Africa, indigenously, there is a possibility that African parasites can develop their own markers of resistance, and that may lead to those phenotypes. And this is the case in Rwanda, this is the case in Uganda, possibly Tanzania and in Ethiopia as well. Now, exploring this genomic data as well, we've asked the questions, if markers develop in one population, can they spread to other populations? Yes, they can. And it's easier to spread markers in the eastern populations. I don't know why the parasites in Eastern Africa are more related than to each other than in Western Africa. Obviously, the argument could be that in West Africa, because of the high rates of transmission, there is a lot of recombination that is breaking down the relatedness between parasites within a short time. But in East Africa, it's quite prominent. And it might be the reason why when drugs resistance was introduced in East Africa, it was easy for it to flow to Central Africa, and then there was a kind of a barrier for it to flow to West Africa smoothly. But it did actually arrive West Africa finally. Now, homing down for us to look at it in country as well, because we have different regions in the country. I'll typify the case of the Gambia. There is SMC being applied in the east and some parts of the west, but not in every region of the Gambia. Now, these differences might be affecting the way the parasites respond to drug. I'm not going to entice anyone here with the nice pictures of the Gambia, because you're already here. I could use this in another talk somewhere else other than the Gambia. Now, there are several projects we are rolling out to further understand this. There are about 5 projects I've listed here, but most of these projects are doing 2 things. One, survey the parasites in the population, sequence them to understand the diversity in their genetic matter, and also, lastly, try to do some phenotypic studies to see whether this variation allows us to get the correlation between or association between the markers and the phenotype. Now, we are lucky to be in the Gambia for one reason. Over time, 25 years, of course, is a long time. We now can actually, together with Dave and collaborators in this. And this is important because we can track the change of the parasite markers that are responsible for their adaptation to our environment as we have been implementing control strategies. We're also collecting parasites across the Gambia. So we have all of these collected across these 50 years. This is one of the oldest studies I did with Dave. I think it's the first one I did when we were not sequencing. We were actually using microsatellites, where the strongest regions of the genome that show selection are those that code for drug-resistant markers. It is the same thing I'm showing here, where you're seeing the evolution of parasite markers increasing in frequency in the population. And this was done by David and Davis, using some of the parasites that are present in the freezers from 1984 up to 2016, or 2008, yeah. And you can clearly see how those markers evolved. And I advanced those studies by comparing 2 populations, the population in 2008 versus the population in 2014. And once again, we can see regions of the genome that are showing strong selection, directional selection, are those that code for genes that seem to be associated with drug resistance. Now, we've combined all of these from 1984 to 2014, and I'm showing you a composite structure of the genome here, where you're seeing several regions of the genome showing potential markers of evolution and adaptation of the parasite in the Gambia. Now, on the right, you can see still those markers evolving over time with the resistant types increasing in the population. Now, I put there an arrow on the amino acid transporter on chromosome 6 that we did not know the role it was playing, but we had seen it in our genetic studies all the time. Each time we do an analysis, we see this peak coming up together with the peak on chromosome 7 that we know is responsible for chloroquine resistance. Now, one of the mutations in that peak, you can see its evolution. They've already showed this over time. And it has been increasing in our population. And the reason why it's been increasing, obviously, together with chloroquine, might be because it's being selected by chloroquine or other drugs. And the frequency of the mutant forms and the wild type varies across populations across Africa as well. So there is an indication there that probably 2 genes located on 2 different chromosomes are generating variants that may be acting together to increase the fitness of the parasite in not only in the Gambian population, but multiple populations. And that seems to be true. In collaboration with other colleagues in Texas, we are doing this work together. And I'm showing on the right side the analysis in the Gambian population, showing how over time, the linkage between mutations in 2 different chromosomes in 2 genes are actually changing. So these 2 genes are working together. And on the bottom left-hand side, it would seem that if you have mutation in those 2 genes coming together, those mutations increase the resistance of the parasite or tolerance of the parasite to drugs. Now, if this is seen phenotypically in the laboratory, you want to check whether actually if you find those variants in the field, those phenotypes can be repeated. And so we have a project funded by the EDCP, I think Michael is here, that is helping us to do this across 3 countries, increasing the capacity to do actually in vitro testing in laboratories in Nigeria, for example, and also improve the kind of tests we are doing by using new methods that use flow cytometry to determine viability of the parasites, as well as tests for new drugs that the pathogen box of the Medicine for Malaria venture are actually developing against the parasites using these methods. Now, what this is allowing us to do is to classify parasites phenotypes against various drugs. And we can show, I'm showing you the right box there, where you can see parasites that are exposed to drugs for 72 hours and they continue to grow. There are few of them, and these are fatal and very lethal concentrations of drugs. They're supposed to kill those parasites. There are just a few of them, but if you do it across multiple populations, you might have a handful of parasites that are showing the phenotype of tolerance that we can now study against the genome to further understand the relationship between phenotype and genotype. And we started this work with one of the PhD students we have in the lab. And what she did here was to test, these were just 56 isolates. So we didn't have the power to actually do what we call genome-wide association analysis. But we attempted genome-wide association analysis, and it did not fail. In which case, even the variance in the phenotype, though it exists, and we are trying to standardize by using new standard ways of making drugs, by making sure that we make the plates and share them, by doing quality control against the concentrations of the drugs, and by sequencing together. We are now proving that it's possible to do genome-wide association studies with these drugs and genotypes. So at the moment, when we did this analysis, we can clearly see that the markers of drug resistance in the genome are those that are associated with in vitro phenotypes. But importantly, we're finding some new markers that, strangely, I don't know if Dave is going to disagree with me on this. We will discuss about this again after this. We're finding a marker that is an antigen, and it's around an antigen cluster. Now, this gene partakes in invasion. It's not expressed by all parasites in a culture. And it's within a cluster of multiple different antigens, and they help in the invasion of the parasite, to the invasion of the red blood cell by the parasites. It seems to strongly associate itself with resistance, but we don't know to which specific drug it might be driving. So we will continue to survey these markers, and there is a project also funded by the NIHR that is helping us to survey these markers across multiple populations in Africa. And we have, at the moment, I think about 6 different countries in the Genomic Surveillance for Malaria project, and we're capitalizing on the various networks we have to also increase the number of countries involved in the surveillance. Now, the MRC is key to this surveillance. It's key because we have the genome platform that helps us to generate data. We are very used to the SOPs, and we also have the capacity to do the analysis due to the servers and also some of those that we are training, like MSc students from UTG and other African countries, that helps us to do the bioinformatics in our facility. Now, this is just to exemplify what this data can do. In most of the genomic data analysis we've done, there have just been a handful of Nigerian isolates. But through this collaboration, we now have 2,213 Nigerian isolates. And here we're showing an analysis of about 1,600 of them, using just one marker that is also associated with the current drugs that are being used against malaria. And you can see on the left side that the heterogeneity in the distribution of this marker in the population. Now, this is work that is done here in the Gambia, while we are sequencing this parasite to further understand what are variation across Nigeria with a high population density. 27% of malaria incidents in the world might be showing something more important for us to take note. This is the last example from Mali as well. We're doing a similar thing. And here you can see spatial changes in the markers as well as temporal changes in those markers. So at the end of the day, what we hope for is to translate this data, and that translation cannot happen without the NMCP. It can't happen without the African Center for Disease Control. And we are now working together to develop a framework for genomic surveillance of malaria across the continent, that the NMCP should own this data. And this is an exemplification or example of what that can derive. With the NMCP, we selected those sites across the Gambia. So because they said those sites are important for them to survey. And we are surveying these sites for these markers and for the evolution of the parasites. But just by collecting parasites from clinics, we can start seeing the trends in incidence of infection in clinics. Some of it that might have been due to COVID or other factors. In places where infections were low, we're now seeing slightly higher infections occurring. And lastly, we can also show with this data how parasites are flowing across the Gambia, which is important for the NMCP. Now, in these analyses, notably is that if the parasites don't carry drug resistance genes, drug resistance markers, they easily cause epidemics within compounds, and you find them across the Gambia more easily than those that carry drug resistance markers. I think this is important, and that's the information we want to translate, and that's essentially the conclusion we have, that the NMCPs must be involved in our analysis. Thank you very much. There are many people I have to thank for this. Thanks a lot. Sorry. Sorry, I had to.
That was absolutely fascinating. What a fantastic talk. Do we have maybe one or two brief questions? I can see one at the front here. Do we have a roving mic? Do you want to, if we don't have do you want to ask your question and I'll repeat it for people?
Thank you very much. I mean, to all the presenters, and Alfred as always. You've talked about the mutations, of course, the adaptations and the parasite over the years in the Gambia. I just want to check, maybe I must have missed that, whether you have seen any differences between, regions within the country, because we know that the transmission, is sort of different between the western region of the country and the upper river region of the country. I don't know if you want to speak to that, please.
I think between the western and the eastern region, we haven't seen difference in the variants, but over time, so the effect of time seems to be stronger, at least within the Gambia, than the effect of geographic separation, because parasites in the Gambia, in any case, they move around a lot. So you expect them to share similar variants. So we haven't seen differences in the variants, but the frequencies, I was not able to show that. Our preliminary data from genomic surveillance here did show that there are differences in the frequencies in the mutations between the east.
Also, one out of curiosity, over the years, I suspect, of course, we have changed treatment regimens over time. I don't know how that relates with the timing of these changes that you have seen over the time, over the period.
I think it's very clear from the analysis we did with A81 that the chloroquine, when it was introduced in the 80s, rapidly selected for the resistant type. Now, with SP, it's the same thing. When SP was introduced, it rapidly selected for the resistant type. When chloroquine was withdrawn, here in the Gambia, there has not been a rapid replacement of the resistant with the wild type like has been shown in other, while we are not using chloroquine. When in Malawi, for example, it essentially went down to less than 20% of the parasite carrying the mutant type when chloroquine was withdrawn. So again, it boils down to the differences in the parasite type backgrounds and the way that they relate with each other in the east and the west. And this is why we need to continue to do genomic surveillance to understand these dynamics in our populations as we change interventions.
Fantastic. Thank you very much indeed. I think probably what we should do actually is close questions now, because I think we do need to move on to the next session. If you've got a question for any of our fantastic speakers, then please do ask them in the break. Perhaps we can give another round of applause for three really excellent. And we'll hand over to the next set of speakers, more on malaria.
Hi, good afternoon, everyone. So, sorry for this. Is it okay now? Okay. Myself, my name is Babacar Faye. I'm a parasitologist, professor at the Faculty of Medicine in Dakar. Sorry.
Controls room, please. The projection on the screens.
So I'm a director of African Center of Excellence working on environment, health, and social science. So I'm very happy to be here, and I want to thank the organizer of this very nice event, MRC, and also LSHTM. So I want to thank Hassan and also Umberto for that. And as we have some trouble with the techniques, so and we are a little bit late, so we start our session, the last session on malaria. So we have 3 speakers, two here in this room, and also another one online. So we start with Prof. Abdesalam Nou, Mohamed Nou. I give you the floor to introduce yourself first. Give us your presentation.
Thank you. I chose to introduce myself because my bio in the booklet is a little bit outdated. I don't work for the Cambry Wellcome Trust. For the last 6 years, I've been working for the World Health Organization and the Nairobi program. So there's a huge element of fond memories and relationship that still persists. I'm grateful to the MRC for inviting me to the Gambia. This is my first time in the Gambia, and I have been asked to talk about the use of routine health information. I assume this is related to the frequent questions that Umberto asks during the MPAG meetings. One of my roles in the WHO is to publish the World Malaria Report. I lead the publication of the World Malaria Report. It's a document many of you reference, but very few of you actually read. In the last 3 days, the document was quoted 3 times. The first time, it was 5 years out of date. The 2 other times, the numbers were misquoted. And it's not a surprise because while it's a commonly used document, it remains a commonly misunderstood document. So I wanted to shed some light on some of the methods we use. There are 3 methods that we use in estimating the burden of cases. I'm not going to go into how we estimate deaths yet because it's even more complicated. And of the 3 methods, one is just the direct use of the data that comes from countries. The second is a parasite prevalence to incidence model that is used in moderate and high transmission settings. The third method is the one that is relevant to the Gambia, which is the adjustment of the routine data to try and estimate the burden of malaria cases and incidence. Before I go into the methods, I'd like to point out, broadly speaking, that we don't think of WHO in our normative guidance is actually the people involved in the system. And I wanted to go back to a point raised by Kara during her presentation, where she talked about the increasing use of digital solutions in strengthening health systems, but also the high failure rate of those digital solutions. And among the many reasons why they fail is that they're not centered on the people aspect of the system, whether it is capacities, acceptance, and many other things that are related to how people use these digital solutions. The second thing I wanted to point out is that surveillance is beyond disease surveillance, as you know, and it involves a number of things. Alfred just presented a huge component of surveillance related to antimalarial surveillance. There is entomological surveillance, there's HRP2 surveillance, there's the intervention surveillance, so there's a whole space of surveillance, some of which come through the routine system, which I'm not again going to talk to today. Another additional point is that malaria surveillance systems are not static. They are dependent on where you are on the transmission continuum. In a country like Gambia, which has very low burden but hasn't yet really reached the end stage elimination, they rely still on aggregate data that comes from the routine system if you discount the information that comes from the study sites. In the far end, in the elimination settings, where the concern is case investigations and active case detection, I wanted to just point out that surveillance systems are dynamic within transmission continuum and across the transmission continuum. How do we use the routine data? Should I wait until this one comes up? I just continue. How do we use the routine data? There are many considerations we make. We start from the assumption that there is a population cohort that is healthy until they get sick. And when they get infected with malaria, a proportion of them will be asymptomatic. A proportion of these will progress to mild disease, and a proportion of that will progress to severe disease, and some will die. Some who are asymptomatic may progress to severe disease as well, or may recover and become healthy. So it is trying to determine the proportions across every single part of this pathway that we need to be able to actually quantify the burden of disease in a specific setting. Now, what are the challenges we are dealing with? First, when we are talking about the overall denominator of population, of course, we need to understand the population at risk. And very often, we rely on census data. The challenge with census data is that many of them are old. Now, if you take the example of the country that contributes the second highest number of malaria cases, that's the DRC, the last census was done in 1984. There are many countries with censuses that are 15 years out of date. There are others that are about 10 years. In the Gambia, I think the last census was done in 2013. And so, because of the infrequent censuses, you have to do projections. And the more the distance between censuses, the more uncertain you are with your projections. Now, when it comes to asymptomatic infection, the only source of data that we have are household surveys. As you know, again, these are done infrequently. Uncomplicated data comes from the routine system if they seek care. But we know there's a good proportion of uncomplicated malaria that don't seek care. Severe disease, the same. When it comes to death, some die in hospital and are reported or may not be reported. Some don't die in hospital. So the challenges we face across the pathways related to the uncertainty in census projections, issues related to the infrequent household surveys. The routine data that we have is mainly from the public sector across all types of routine data. And some of the issues that you have to account for in the process is really related to, for example, when it comes to community surveys, many countries do their community survey in part to be able to measure intervention coverage. So they do a mass campaign, they do a community survey a few months shortly after, and they are trying to tap into the peak, inadvertently, into the peak effectiveness of that intervention. They see a dip in prevalence. They think that dip in prevalence is a signal of dip in disease, which isn't the case, because the effectiveness of that bednet wanes, and 2-3 years down the road, you see your incidence levels haven't changed much. The second problem we have is they come from limited age range. However, they do provide us information on asymptomatic data. They also provide us some information on treatment seeking. With the routine data, because the data comes mostly from the public sector, not all of them report, not all of them are confirmed, and some of the reports of positive malaria may just be incidental parasitemia, and the actual cause of febrile disease is another disease. And we rarely have any information from the public sector, private sector, or those who don't seek care and stay in the community. You have all these biases running through the data sets, whether it is uncomplicated or severe disease and deaths. And when it comes to death, we know, as you can tell from your DSS surveys across many sites, that a lot of mortality occurs at home and is barely recorded. So how do we describe this whole complexity and try to capture empirically in the quantification of disease? And I want to credit my colleague Beatrice Galatas for this figure that I'm about to display. Now, the data that WHO receives, or the data that most of us have access to, comes from the public sector, as I said. And you have the total outpatient patients from those that have reported. Within that, you have those who have been suspected of malaria, the tests. Within that, I said the tests, not all suspected are tested, not all outpatients are suspected, and not all those who are tested are confirmed. But you have a challenge. By the time you are testing and deciding on whether the person is positive or not, there's a proportion of people who are suspected who you haven't tested, who could have been positive, and there's a proportion of patients, outpatients, who you didn't even suspect, and therefore you haven't tested, and who could be positive. So the routine data itself, even if you have high levels of confirmation, may be incomplete. And then to add to that, you have a problem of when you relate the confirmed, the positives reported to the treatments, there's a discrepancy because a lot of negatives are being treated presumptively or a lack of compliance with the RDT test. To add to that complexity, we have only a subset of those who are reported, and then we don't know who seeks care in the private sector, and we don't know who stays at home and doesn't seek care. So it's these complexities that we try and figure out in the World Malaria Report, and it's a complexity that you need to figure out if you are to use suboptimal routine data to make decisions for national strategic development and planning. What does this then mean in terms of methodological approaches? I'm not going to go into all the equations, just to say that you start with the crude reported data, you adjust for testing rates, then you adjust for reporting rates, then you adjust for care seeking, and you have an estimate of malaria cases, and you convert that into incidence using your population denominator. There are many benefits to this approach. It's stepwise. It accounts for some of the systemic challenges. You can account for what is in the public, what's in the private, but it comes with a number of uncertainties because there is uncertainty in the parameters that are involved in the estimation. What does this mean for the Gambia? If you look at the red line, since 2008, when Gambia started reporting cases to the WHO, what you see is in the red line what's been reported through the system, slightly under 100,000 cases. What you see in the green line are those cases from the public sector after you've adjusted for testing and reporting, which essentially indicates that the testing rates and the reporting rates have been gradually improving. Now, what you see in the purple line is when you start adjusting for the private sector use as well as those who may not be seeking care in the public sector. And so you see the biggest determinant in the difference between what's been reported in the public sector and what's been estimated in WHO, which nearly doubles the cases, or more than doubles the cases, is related to the private sector and those who don't seek care. So that's one approach we use in the WHO. A second approach, which is a little bit more sophisticated, is work we've started with the Malaria Atlas project, where we start with the assumption that if you have case data from health facilities and you have community parasite prevalence data, even though the two are not necessarily the same thing, that there is an epidemiological relationship that you can model. The beauty of the community parasite prevalence survey data is that they are robust. They do not suffer some of the biases of the health facility data. The challenge is that they're infrequent, that they have a poor temporal signal, and they do come from a specific age range. The benefit of the routine data is that they have a very strong temporal signal, and they do cover a wider age range. And so if you try and develop a relationship between the two, you can actually develop a better measurement of incidence and a better measurement of prevalence. And so if you take the example of Nigeria, where we start with just the reported cases and an estimate of parasite prevalence, you take these through a whole series of modeled interventions, as well as malaria interventions, as well as climatic covariates, you come up with a predicted incidence, in this instance, in children under 5, but you could do the same map for prevalence. And what we have noticed is that when you do the out-of-sample testing of either highly controlled routine data or parasite prevalence data, you do get better reliability in the joint modeling of parasite prevalence and incidence. The challenge, of course, they are not processes that are readily available to countries so that they can use for their frequent response and strategic thinking. And that's why, as WHO, we recommend the simpler approach to help countries use the data for decision making. Now, to end my presentation, I'll say that at any stage of the transmission continuum, whether you are in high burden or in elimination setting, your routine passive case detection platform is the backbone of your malaria disease surveillance. There has been a tendency, unfortunately, over many years to ignore the passive case detection system and invest in community surveys in order to measure the impact of donor investment. I think there has been a realization, again, over the last 5-6 years, that that is not sustainable, nor does it give you all the information you need, and now there's increasing investment in supporting routine system. However, the gap is still massive. The second point is that if you are going to use suboptimal routine data to make decisions, you have to look out for a number of important factors. You have to account for low access to care in some settings. You have to account for inconsistent pathological diagnosis, weak adherence to tests, low reporting rates, inaccurate reporting, weak epidemiological investigations because of the poor age structure in the data, as well as some countries that do not have the benefits of research-based epidemiological investigations that help you understand the age patterns and other factors. There is a problem of the unreliable definition or adherence to the definition of severe disease, severe malaria case definitions, which affect the cause of death fractions, and then overall weak mortality reporting. The third aspect I want to talk about, which I mentioned at the beginning, is related to digital solutions. I am a big supporter of digital solutions. They do offer enormous efficiencies in our surveillance systems, but by focusing just on the digital solutions and ignoring the other important systemic aspects, particularly the human capacity, we're just going to see a series of expensive failures. And finally, I would urge that despite all of these challenges, we should see them as opportunities to solve problems, and we should continue to use the data to make decisions at the country level. Thank you very much.
Thank you very much, Prof. Noor, for this very interesting presentation. So we'll go through the program. We have now an online presentation, I think so. With Professor Clara Menendez from Barcelona. So do you have the presentation, or she's online? Okay.
Yes, I'm online.
Okay, okay. Nice to hear you. So Prof. Menendez is an epidemiologist working at Barcelona. So she's a specialist on child and she leads the maternal and child reproductive health initiative, and she's also just a member staff of the International Health Department at the University of Barcelona. So Prof. Menendez, I will give you the floor for your presentation, and just after we will have a question for you as you are online. And at the end, we will have the question for the two presenters, speakers here with us in this room. So you have the floor.
Thank you very much, and good afternoon. First of all, I would like to thank Professor D'Alessandro for inviting me to this very important celebration of 75th anniversary of the center in the Gambia. And I'm very sorry not having been able to be with you there. Another thing is because in the Gambia, I started my professional career actually in Farafeni, working with Professor Brian and Professors Brian and Alice Greenwood in trials of malaria prevention in pregnant women. Let me go to the next slide, please. Just 1 second, please, because I have problems to go to the next slide. Where did you go? Okay, so I started working in the Gambia in these projects and clinical trials of malaria prevention in pregnant women. And one of the I think, pioneering things that we did at that time was to train the traditional birth attendants of the North Bank primary health villages to collect placental tissue biopsies and to prepare thick blood smears from placental blood in order to assess malaria in the placenta and therefore in pregnancy. And here there are two pictures of some of the workshops, training workshops that we organized also to show our gratitude for their high commitment, and without them these studies were not possible to be done. Let me then go to the more specifically to the talk. So drugs for malaria prevention have been used for a long time and in different ways. And I mean, from the days of the first malaria eradication campaign, you can see here how quinine is handed over to a woman by a nicely dressed, of the time, lady field worker. Of course, in Africa, in many endemic countries in Africa, chloroquine was regularly administered to school children to prevent malaria in the 60s and 70s. So this has been done already. But where is malaria prevention? Sorry, let me go again to this one. So we have different ways of understanding chemoprevention to prevent malaria. We can use chemoprophylaxis as it was is defined as the regular administration of an antimalarial drug, and this has been done in pregnant women and school children, as we've shown, and still done in travelers to endemic areas. And we have used chloroquine, proguanil, brimethamine, atovaquone, plus proguanil these days. And we have shown, it has been shown, that chemoprophylaxis reduces clinical malaria, it reduces anemia, and even all-cause mortality. And this especially from studies, large studies done in the Gambia some years ago. On the negative side, it increases potentially resistance to antimalarial drugs. It has cost implications. And there is the question of what is called rebound, which is the potential increase in the incidence of malaria once chemoprophylaxis stopped. There was on this particular issue, there was a technical consultation carried out by or organized by WHO last March. And the conclusion of this consultation was that, based on the evidence, it doesn't appear to be a frequency or magnitude to warrant serious concerns about current interventions. So, what are the malaria chemoprevention strategies currently recommended by the WHO? Well, one of them is seasonal malaria chemoprevention, which is called SMC. This was recommended as policy by WHO since 2012. It is recommended for highly seasonal transmission areas, has been quickly adopted in these areas. The approach of the delivery of the strategy is a delivery through door-to-door by community health workers, and is given 4 times in monthly rounds during the transmission season, of malaria transmission season. It is usually based on sulfadoxine pyrimethamine and amodiaquine, and this is a 3-day regimen. The main challenges that this strategy has are the sustainability, because it has to use a not always existing or functioning program, that of community health workers. The compliance, these 4 times coverage varies widely from 45% to 88% of eligible children receiving it. About half of them are of the children who are eligible don't receive it, according to some estimations, and it has clearly cost implications of obviously paying the community health workers and their training and the supervision and the moving around during the transmission time. Then mass drug administration, MDA, is also recommended as a WHO policy since 2017, and basically consists of the administration of an antimalarial treatment to all age groups of a defined population. It is recommended either for burden reduction, but this is in high, moderate P-falciparum transmission areas, and especially during emergency situations, or for transmission reduction of in very low or low transmission areas of P. falciparum and of P. vivax. And it has the advantage of reducing transmission quickly and also the burden in these settings. On the challenges side, the effect that is achieved both in, or either in transmission or in burden reduction tend to wane within 1 to 3 months, and therefore needs to be combined with other effective malaria control strategies. And they are also not so well recognized drug-related safety issues, specifically those related to women of childbearing age, because which correspond to about 20% of the population at one point in time in most settings. And these women may be pregnant. About 30% of them actually are in the first trimester of pregnancy, and so that depending on the drug that we use for mass drug administration, we can get into not well recognized and not well accepted drug safety problems. Then the other chemoprevention, malaria chemoprevention strategy recommended by the WHO is intermittent preventive treatment. This consists of the administration of treatment doses of an antimalarial at predefined intervals. And this is given irrespective of the presence of parasites, although it's called treatment. It's more prevention, but we call it treatment. Makes use of existing health schemes, either antenatal care clinics or the API scheme, and is currently only based on sulfadoxine pyrimethamine. We have two types of intermittent preventive treatment that relates to pregnant women. It's called IPTP. Policy has been policy by WHO, recommendation as policy by WHO in areas of high to moderate transmission since 1998. Has had a slow adoption. As I said, is delivered through the antenatal clinic, but also more recently, it's been accepted to be delivered also by community health workers in addition to antenatal clinics. It has been shown to reduce neonatal mortality, which is an important clinical and public health outcome. There is no evidence that it causes or is associated to an increase in drug resistance, and it has been shown to be effective even in areas of moderate to high sulfadoxine pyrimethamine resistance. The challenges are, well, the coverage is still very low. In last year, over 30%, only 30% of eligible pregnant women receive at least 3 administrations of the monthly administration that they should receive. This may be related to health system barriers and barriers that we still don't know very well. And there's the challenge of what to do in the context of HIV co-infection because of different drug interactions that I will talk about in the next slide. And there is also the issue, the reproductive safety profile of the new alternatives coming, since we have observed in different countries an increased proportion of women attending the antenatal care clinic in the first trimester. So we need to do something about those women coming early in the pregnancy, as we recommend them to do. So just a few words about the problems of malaria and the challenges of malaria prevention in HIV-infected pregnant women. This is probably not, fortunately, it's not a huge problem in West Africa and the Gambia, but it is in South Africa and East Africa, where we still have about, and altogether in Africa, they're estimated that about 1 million pregnancies are complicated by co-infection with both malaria and HIV annually. So these are recognized as the most vulnerable group to malaria infection, because we have observed that there is an increased risk and severity of the infection in pregnant women who are HIV positive. In them, the efficacy of antimalarial drugs is reduced, probably because of the lack of the immunosuppression. And there is also an increased risk of mother-to-child transmission of HIV when they have malaria. And at the same time, and paradoxically, that's what we published this paper some time ago to advocate and attention for this, they are at the same time the least protected. Why? Well, because sulfadoxine pyrimethamine, which is a recommended drug in intermittent preventive treatment, is contraindicated due or for fears to drug interactions with cotrimoxazole prophylaxis that they need to receive to prevent infections. This cotrimoxazole, although it has some antimalarial effect, the protection that it offers is only partial. So what studies have been done on this issue, on this problem of malaria prevention in this particular group? Well, not many, actually. We did one using an alternative drug, mefloquine, some years ago in Kenya, Mozambique, and Tanzania in over 1,000 pregnant women. And we observed that mefloquine, which is that doesn't have interactions with cotrimoxazole, so therefore could be given, was associated with significant reductions in maternal parasitemia at delivery, placental infection, and even, and importantly, hospital admissions. And there were no differences in terms of safety, in frequency of poor pregnancy outcomes, or serious maternal adverse events related to medication. However, we observed something unexpected, which are not the vomiting and dizziness, this was expected, but we observed a significantly higher HIV viral loads at delivery and significantly higher rates of mother-to-child transmission of HIV in women who had received mefloquine for malaria prevention as IPTP. So therefore, this drug could not be recommended for to replace to add on to cotrimoxazole in these women. Another small trial was carried out recently using dihydroartemisinin and piperaquine, ACT combination in Uganda. A small study that didn't have enough power to see a difference in malaria or clinical outcomes. Now there are ongoing 2 clinical trials, randomized controlled clinical trials on this question, to solve this question, or trying to solve this question. One is carried out by our group in Mozambique and Gabon in over 600 pregnant women HIV infected on cotrimoxazole prophylaxis and antiretrovirals. So we are looking at drug-drug interactions in this trial. Similarly, there is another one very similar in design that is carried out at the moment in Kenya and Malawi in nearly 900 pregnant women, again also on cotrimoxazole and antiretrovirals. And I think by the beginning of next year, we will be able to provide conclusive information. And I think having these 2 trials with very similar designs would be very good to provide to WHO some results that they can take on the recommendation or for malaria prevention in this group. The final strategy of malaria chemoprevention is what we call, up to very recently, intermittent preventive treatment in infants, but now it's being renamed to perennial malaria chemoprophylaxis. And I will tell you in 2 minutes why this has been changed in the name. This is policy WHO recommendation since 2010, is delivered through the EPI scheme, and very slow, or more than slow adoption. It's only been recently adopted in Sierra Leone as national policy since 2018. It's been recognized as highly cost-effective. The new challenges since it was recommended in more than 10 years ago is, well, the EPI program is moving to other schemes, such as the outreach EPI clinics. There are more outreach EPI clinics in some countries than the fixed clinics, and this needs to be taken into account when delivered this intervention. Also, we in these years, we are realizing that in some regions there are a number of children who are born, or they are HIV positive, or are born to HIV positive women, and they are also on cotrimoxazole prophylaxis themselves. So sulfadoxine pyrimethamine cannot be given to these children. So we need to look for alternative drugs for this group. New vaccines have been added to the EPI program, pneumo, Haemophilus, rotavirus, and we need to rule out potential vaccine interactions. And then finally, and very importantly, we need to have a good pediatric formulation, such as dispersible SP tablets. More than 15 years ago, we evaluated in the context of a consortium, an international consortium funded by the Bill and Melinda Gates Foundation, called the IPTI Consortium, we did a comprehensive evaluation of this strategy that went from molecular biology studies and immunological studies and interaction, vaccine SP interactions, to the cost effectiveness evaluations and acceptability studies through clinical trials that were carried out in 8 countries in West, East, and Southern Africa. There was a pool analysis of 6 randomized controlled trials, the 6 ones that used sulfadoxine pyrimethamine, that was published in The Lancet in 2009. And we found in this pool analysis significant reductions in clinical malaria by 30% and 75% reduction if we look at the 35 days within administration of the drug, which is the duration of the efficacy of the drug. There were also reductions, and this is worse. These reductions were looking at the effect at 12 months of age, but the drug was given, the last dose was given at 9 months, so 3 months, between 9 and 12 months. And there were reductions also in hospital admissions with parasitemia and all-cause hospital admissions. More than 10 years from then, I mean, at that time, in 2010, when WHO sulfadoxine pyrimethamine was not high, that is, that quintuple, and that was defined as the quintuple mutation to this drug being less than 50%, and co-administer these vaccines. The new guidelines on IPTI were released this year. I think it was April or March, if I'm not wrong, and they changed to these 2 very important changes. It is now recommended for 2 areas where sulfadoxine pyrimethamine is the expansion of administration into the 2nd year of life. And that's why I come now to the new name. It's now called perennial malaria chemoprevention, PMC. So we need to get used to this new acronym. It still is one of the most cost-effective strategies for malaria control because it relies on an inexpensive drug, sulfadoxine pyrimethamine. The full regimen is less than $1, and using an existing health system, which is the EPI system. That helps in the susceptibility of this malaria control strategy. However, to date, it's been implemented nationwide only in Sierra Leone. That is why we think, we believe, all the investigators have been working on this for many years, that we have to avoid another lost decade in reducing malaria burden in African infants and young children because we have the tool to do that. There's a momentum around perennial malaria chemoprevention in the sense that in partnership with ministers of health of African countries, there's been in the last couple of years an increasing interest by different research and development stakeholders, from the ADCTP to the Bill and Melinda Gates Foundation, UNITAID, and some private and development cooperation from different countries to invest in this strategy so that it is to facilitate the implementation by the countries where it should be implemented. So just a few words about one of these initiatives is the Multiply project. It's an implementation project using the EPI platform, but not only the fixed clinics, also the mobile outreach clinics. And the aim is to expand in the areas like Sierra Leone. This is carried out in 3 countries, Sierra Leone, Togo, and Mozambique, and to expand the implementation of IPTI in Sierra Leone, which is only 3 doses, to 6 doses, and to start and facilitate the implementation in the 2 countries in children under 2 years of age. And we aim to implement this intervention in more than 45,000 children. I'm about to finish just to explain a little bit how what is this the changes between IPTI and perennial malaria chemoprevention. So if you look at in this graph, the IPTI, what it says IPTI is there the currently recommended, or up to now, it's been recommended and implemented in Sierra Leone, IPTI as the intervention with SP at 2.5, 3 months when they come to receive the DTP1 DTP2 and 3, then measles, and that's it, three doses in the first year of life. Where we want to take the advantage of the system, allowing for more contacts, and we're going to use also the vitamin A supplementation contacts that in some countries are given at 6 months intervals, and also the booster measles given in between 15 and 18 months of age. So altogether, there will be between 4 and 6 more doses of protection of this intervention. Just last words about drug resistance that has been mentioned before. Drug resistance in chemoprevention cannot be isolated from in vivo studies or treatment studies even with the same drug, because although, of course, chemoprevention may lead to increased prevalence of resistance genetic mutations, there are many factors apart from the resistance of the parasite resistant to the drug that affect the efficacy of the strategy, and the most important one probably is the naturally acquired immunity. This is given to people who are asymptomatic and most of the time they are semi-immune. And we have seen that even high rates of resistance to the drug, in this case SP, don't impair chemoprevention efficacy. It's important also to recognize or take into account that these drugs are only used for prevention, and they target a small proportion of the population, about 10% between pregnant women and children. Also to highlight that there are no marker or set of markers, although we are looking for them carefully, that reliably predicts the outcome of a regimen given in an individual for prophylaxis or chemoprevention. So the decisions in terms of what to do, in terms of how to work to implement these strategies still need to be made on the basis of imperfect evidence. And we still need to continue monitoring patterns of resistance, which can help guide policy recommendations, but they are not the only factor that should be taken into account. The WHO, just to finalize, has recently, I think also last August, developed simplified protocols to evaluate the ability of chemoprevention to clear in terms of efficacy and impact. And I think this is my very last slide, just to show how we have to see prevention in the most vulnerable population as a continuum of prevention, more than continuum of care, from the pregnant woman with given intermittent parenteral treatment, to the young child, to the older children with SMC. And I think this is all from my side. So thank you for your attention. Thank you very much.
Or do you want to receive now your questions? You have only one presentation remaining. So you hear me?
Yes, I can hear you. Yes.
Yeah. Do you want to stay with us until the end of this session, or do you want to receive now your questions?
Well, if I can, well, whatever, I don't mind.
Okay, maybe we can continue, and after we'll have a Q&A session. So I will give the floor to the last speaker, Professor Annette. Nice to have you on board on this session on malaria. So she's coordinated the MRC Malaria Research Group, and she will give us an overview of the research on malaria in the MRC. So Annette, you have the floor for your presentation. So maybe we can have questions for Clara and so Professor Noor before having the presentation of Annette. So the floor is yours if you have questions. Yeah, I have three.
Hi, I have one question for Professor Noor. Really amazing presentation. I was really perplexed with, how do you adjust for the cases that are going to private health facilities? I think that's bad. And I had a question for Clara. So this is Kamija from Malawi. Great presentation. I was wondering, I know you mentioned that IPTI SP can be used in areas where there's not high resistance. And I was wondering whether you had any thoughts about what drugs could be used in such areas where there was high resistance. Obviously, you take into account the first-line treatment or drug of choice that's being used in such areas. But some thoughts on that would be great. And lastly, I noticed that you did not mention in your chemoprevention strategies post-discharge malaria chemoprevention in conflict of interest, but it would be great to hear what your thoughts are. Thanks.
Well, I'll quickly respond to your question, Firi. So the only mechanism right now to adjust for care seeking in different sectors is by relying on the household survey information on where children with fever go for care. There are two challenges with that. The first is that it's only in children, and we assume it's representative of all ages. The second is that we implicitly assume that the probability of disease in patients who seek care in the different sectors is the same. And that's why if we could get information that actually looks at the pathway of care in much more detail, because there's some people who start from the kiosks, go to the private clinic, then go to the public health facility. If we could understand that pathway across a wider age range, then that will increase things. But the most sustainable solution is to actually engage the private sector in the surveillance system and improve the broad surveillance system in both the public and the private sector.
Thank you very much. Clara, you have a question from Kamija.
Yes, no, thank you, Camilla, and no, thank you very much for your very important questions, actually. I mean, the first one is difficult to respond to. We're still struggling to find an alternative to SP in men and women. In children, again, it would be the same. And not only for areas of high SP resistance, but also in areas of high HIV prevalence, as I mentioned. One alternative that we're still under discussion is amodiaquine, because there is a pediatric formulation, and it's used, as you know, in SMC, as co-package with SP. So this could be an alternative, but it needs to be evaluated, I suppose. As I said, we're still thinking on alternatives to that. The thing also is that any alternative we find, it would be one, more expensive than SP, than the current one, and two, it will be more complicated to deliver or to ensure compliance, because SP is supervised one single administration, while any other regime requires 3 days of administration. So unless we, short this in an empiric way, it would be difficult to ensure compliance. With regard to post-discharge malaria chemoprevention, well, that's interesting because we work on my group and with David Schellenberg from that was working with us in Ifakara at the time. We did some very preliminary studies and trials on post-discharge malaria prevention. And yeah, I don't know, I will ask Noor, Professor Noor, if this is already a WHO recommendation or not. I didn't know, that's why I didn't include it in my presentation. If you allow me, I will also answer one question that I see in the chat about what did we do, and I think it's an important question. What did we do in the trial using mefloquine, where we found an increase unexpectedly, because we expected the opposite actually, an significant increase rate in mother-to-child transmission of HIV. Well, that was very unfortunate. We informed, obviously, the mothers, the participants, and we ensured the provision, because of those days that was not ensured in the country, in Mozambique, the provision of antiretrovirals to their children and to them. That's the only thing we could do. Thank you.
Yeah, just a quick question, a very quick question. SMC has been implemented a decade ago, and over that decade, a couple of countries have been implemented it, and now malaria has gone very low. But still, we are still implementing SMC. So maybe we need to think now, rule how to stop when, obviously, in certain settings, this is not needed anymore. So this is a comment. We need to start thinking how to stop, and how to stop, and what are the indicators that trigger the stopping of such such an intervention.
Thank you very much. Ambrose?
Thank you very much, the presenters. And Noor, I mean, definitely the World Malaria Report has improved very, very drastically over the last couple of years, thanks to you. My question is, in your adjustment, as we get data on the frequency of the estrogen-rich protein-based gene deletions in those RDTs, is it your idea that you try to adjust for this? Because different countries now are beginning to collect this data on a routine basis in different sentinel sites. And so these gene deletions result in false negatives. And so there might be a need to, especially for very, very highly endemic countries, to see that we can begin to adjust for that as well. And the issue that really Dr. Kamija has raised is, which we have really struggled with, trying to improve the collection of routine data from a few private health facilities. And I'm talking about private, not private faith-based, but private for-profit facilities. Is maybe if we could just designate a few, and then you use that to adjust, because we would have some data from the private health facilities, and then you include it in your model for adjustment. But we really struggle with trying to get data from the private sector since we started implementing IDSR in 1998 in these countries in Africa. But generally, thanks for a very good malaria report these days.
Thanks, Ambrose. Just to respond to your last point, the struggle with the private sector continues. Many things have been attempted: regulation, incentivization, engagement, but nothing has proven to be successful. The only success has been realized in the end stage of malaria elimination, when by regulation the private sector have been given the permission to diagnose but not to treat. And so they refer, and by referring they inform the public sector. So I mean, it's still a major challenge, but it's something that needs to be tackled. Now, in terms of adjustments for HRP2 deletions, the data isn't sufficiently, is not available across many countries for us to be able to adjust credibly. It's still very few sites, mixed results. In the country where HRP2 deletion has been a massive problem, Eritrea, they have already switched tests, so the adjustment is not particularly important, except for the years when they had the deletions, which we are trying to account for. And so that's the response to it. Now I think as the data increases, we will start considering adjustments for deletions before countries switch to other tests.
Yes, my question is for Professor Menendez. This is Menno Smit. Thank you very much for your presentation and your effort to provide malaria chemoprevention for this vulnerable group. My question kind of follows that of Professor Kamita Firi in terms of the choice for the drug regimen and also plans to evaluate the modified IPTI or now PMC schedule, which has been expanded from 3 to 6 doses. Have there been any thoughts into comparison of alternative drug in SMC in Southern Africa and pursuing alternative drug resistance, alternative drug drugs for SMC in Southern Africa due to SP resistance? So have any alternative drug regimens been considered, and have any consideration been given to efficacy trials comparing these regimens and also the...
Well, again, thank you very much for your question. I'm not sure there are specific thinking about doing clinical trials, specific clinical trials on different drug regimens for a PMC. What I'm saying is that, what I said before is that I think there is something that we need to look at into because not only in areas of high drug resistance, by the way, I didn't go in detail in my presentation, but there, as I said, there is not a clear relationship with the chemoprevention strategy, in this case, IPTI. In fact, we have from clinical trials in the IPTI consortium information that suggests that this relationship is not straightforward at all. And there are areas with moderately high levels of SP drug resistance, looking at genetic mutations, over 60%, with 75% protective efficacy in the 35 days within the SP dose given. So this relationship is not clear, and it should not guide, as I said, when, or not at when, sorry, if to give SP or not, because it would be wrong. However, it's more important to me, at least, to think on alternatives, because SP cannot be given to children who are contraindicated to give SP. So I think we need to look at into alternatives always. So this is something that has not been, as far as I know, and there is a community of practice on IPTI or PMC recently created of many other groups that are working on this at the moment and in facilitating the implementation in different countries. And this is something that we have recently started discussing, because, I mean, you see, this intervention has not been implemented for the last, since it was recommended 12 years ago. So we start now saying, well, maybe you should not give it here or there or that. We lost another decade. As it happens with IPTP, then we can look at into other things. Just to say that SMC with SP and amodiaquine is given, is now being implemented in northern Mozambique by Malaria Consortium, and in areas of very high SP resistance measured by genetic mutations. And it seems to work. So, I mean, I leave it there because, of course, I mean, my short response would be, we need to look at into alternatives, yes, but this should not prevent the implementation of this intervention. Thank you.
Very last question.
Thank you. If he has time, I just wanted to ask another question for Clara. Clara, this is clearly the concerns for pregnant women and also the infants to receive the prophylactic medication. It occurred to me whether you and the malaria field is also thinking about pregnancy registers and the way that potential, drug interactions, etc., are monitored by public health systems in a sort of coordinated way. I know we're spending a lot of time on this thinking about the implementation of the vaccines, and I therefore wondered whether there is already similar thoughts also in the malaria field, and probably I should know about that being a pediatrician. But if you could shed some light on the deliberations, whether this is very much siloed or is also integrated with EPI and MCA and registration and all of those things to capture the ongoing and potential new interventions in a more systematic way, because we could then join forces. Thank you.
Well, thank you very much. I think it's a very, very important comment, and it needs to be taken into account, especially as you said, with the new interventions and immunizations in pregnant women. Absolutely, fully agree.
Let's talk.
Thank you very much, Clara. So we'll give the floor now to the last presentation of this session. Annette, you have the floor.
Good evening, everyone. Okay. Good evening, everyone. Can you hear me? Yes. Thank you for staying until the end. The good news for me is you have exhausted your quota of questions, so no more questions for me. I'm humbled to close this prestigious event with so many distinguished speakers. So I'm just trying to not put you more asleep and give you a quick overview of malaria research in the Gambia. So as we all know by now, it all started in the 50s with Sir Ian McGregor, and you see him with his wife in Keneba while they were doing some malaria survey. And at that time, as you remember from the first talk, malaria prevalence in children was near to 90% and 50% in the general population. So now, 70 years down the line, especially in the year 2000, 2010, and actually in the past 20 years, malaria has decreased a lot thanks to so many interventions for which the first trial, almost all of them were carried out in the Gambia, and these are just some pictures. The first one is in Farafeni with Brian, of course. The second one is the evaluation of the bednet program by a famous Italian singer or researcher, Umberto. Then SMC, and finally also the first trial with ACT was also done in the Gambia. And also, of course, malaria vaccines are there. So these are data that were graciously communicated, shared by our colleagues from the National Malaria Program, and I would like to thank them to be here this afternoon to look at our presentations. You can see the evolution of malaria incidents per thousand over the past 6 years. And you can see that the two nadir in 2017 and 2019 were actually when long-lasting insecticidal net were distributed massively in the URR and also CRR. And then you see a small rebound effect in 2020 and 2021 during the COVID pandemic. And in red, the red line, so not the same numbers, but the same trend over the years. And when we look by age group, so in the upper 5 and under 5, you can see that in the 2 past years, malaria, number of malaria cases in children especially has increased. So this coincides with the COVID-19 pandemic, and the National Malaria Control Program takes this seriously. These are data over the years, but by region. So you can clearly see that we have two main regions where malaria is still a problem, which are the two extremes, the Upper River region and then the West Coast. The goal for us now is to eliminate malaria by 2025, which is not that far, so we keep optimistic. The whole spectrum of the transmission cycle from parasites, so like basic science in the lab, from genotyping and sequencing, through epidemiology, to mosquitoes, and also environment for area. Sorry for those who have seen these slides, but I thought we should still show our faces to those who haven't seen us. So, of course, Alfred, everybody knows Alfred, and there is also Mamadou Usman. Epidemiology, it's me and Umberto. Then on entomology, we have Musa, who has been there since many, many years. He's our senior entomologist. And we have Aruna. And finally, in social science, we have Fatou Jaite. You see her on the picture. She is finishing her PhD with Professor Koen Peters. And hopefully she will come back to work with us in the Gambia for her, then the impact on the malaria risk. So the objectives is, of course, to contribute to malaria local capacities. So we have several objectives by teams. On the side of the parasite, as Alfred already explained, we are concentrating on genomic surveillance of malaria parasite across Africa. And we also try to understand human genetic factors for susceptibility. And of course, as you have seen with Alfred, we monitor anti-malaria drug resistance with the National Malaria Control Program. Risk factor for residual transmission, and also evaluate efficacy of new strategy, tools, and drugs as we will fight resistance. And they evaluate new insecticides, and of course they are trying to develop new tools to try to optimize the effectiveness of existing or new strategy. And we hope we can also work on climate change. These are just some names of projects that are ongoing or projects. This is, for example, what we call INDI project, which stands for Infection, Dynamics, and Transmission to Inform Elimination. So this study is actually a pilot intervention studies where we have 3 intervention and 1 control arm, but only 2 villages per arm. It's a pilot study. And it's mainly to estimate the effect of different intervention on the human reservoir of infection by looking at the parasite carriage, gametocyte carriage, and especially infectiousness of infected people to mosquitoes. So you see the picture of the junior scientists who are leading this project. Abdullah Ahmad, who started it, and now he moved on. We have Aruna, who is doing the entomological part, and then Bakari Conte, a Gambian scientist, who is now leading the epidemiological part. And this graph shows you the result of the first 2 surveys. I think maybe
you are
not muted the microphone. So on the graph, you can see the 8 villages on the x-axis, and then the prevalence by PCR, malaria prevalence by PCR for 2 surveys during the transmission season in 2019. And this gives you a good a summary of the situation. As you can see, malaria is now very heterogeneous. You can see the prevalence ranging from less than 5 to about 30% during the transmission season. And then the second interesting aspect is that children below 5 are no longer the main group of infected, the main infected group. It's rather older children and even adults. And then the other interesting finding is to see that actually most of the infections were very low, had very low parasitemia. 61% had less than 1 parasite per microliter. If you add the ones between 1 and 10, you are almost at 80%, which means that these are infections that are very hard to detect by standard methods. The infectiousness results shows that, okay, the study is, as I said, is a pilot study. So we found 8 infectious individuals. They were all asymptomatic. And then when we looked at their infectiousness to mosquitoes using direct membrane feeding assays, DMFA, you can see in the second table that the proportion out of total asymptomatic infections, so means that 90% of the infections to mosquito were among people with low parasite densities. So people with low parasite densities are transmitting to mosquitoes, and that's our problem. Another study, probably most of you have seen the result of this cluster randomized trial called MASSIVE project. It has been already presented in different meetings, but I just wanted to remind it for those who would not have seen it. So 32 clusters of villages were randomized to either ivermectin plus dihydroartemisinin piperacin, which is an ACT. And of course, the standard interventions were also implemented in this group. And then in the control group, it was just the standard interventions, meaning IRS, LLIN, long-lasting insecticidal treatment, SMC, and IPTP. On the right-hand side, you can see that ivermectin was given at a dose of 300 micrograms per kilo per day plus DP. And then it was directly observed, and they did 3 rounds, July, August, and September. These are the outcomes. So there were clinical outcomes and parasitological outcomes, as well as entomological outcomes. I'm not going too much into the detail. This is the coverage. You can see it was around 80% the second year. And then the results showed that there was a very strong effect on malaria prevalence. So by the end of the intervention in 2019, the odds ratio, the adjusted odds ratio was 0.28, meaning a decrease of about 70% of the odds of infection. Same thing was observed on the incidence of clinical malaria. The blue line shows the peak of malaria in the control group, while the red line shows the incidence in the intervention group, and this amounted to a decrease of 80% in clinical malaria. And the intervention was safe and well tolerated, and had also a significant effect on mosquito density as well as mosquito mortality. So this was published in The Lancet, and the 2 main authors, Edgar Dabira and Aruna Soumare, are here on the picture. Both of them are PhD students. So we also do individual randomized trial, like DTACT, which stands for actually triple ACT. So as Alfred mentioned, we have now the problem of resistance to artemisinin that is emerging in Africa. So one of the strategy is to use triple ACT instead of a standard ACT. And so this trial compares two regimens of triple ACT versus the standard ACT. If you want more detail, you can contact Edgar. And we are also, this other individual randomized trial is called PIRAPREG, and this is actually testing Piramax. So the ACT is pyronaridine artesunate, which is a new ACT. It has been proven efficacious and safe in non-pregnant adults, and now we are testing it in pregnant women together with dihydroartemisinin in piperacin. You see the three arms, and artemether lumefantrine, which is the first line treatment. We do 63-day follow-up to estimate the efficacy and safety in pregnant women. Okay, now we are also doing studies with the National Malaria Control Program, and these are studies on surveillance of drug resistance. We do also insecticide resistance with them, but I'm now showing the drug resistance data. So the graph shows the previous result found when we do therapeutic efficacy study on artemether lumefantrine, which is the first line treatment. And as you can see, it has remained above 90%, which is good news. But the last time we did it was 2018. So during COVID, we didn't do anything. No, we didn't do this. So now it's high time to check where we are. In terms of resistance to the first line and also the second line, which is dihydroartemisinin piperacin. So we are looking, of course, at the efficacy, we are looking at the safety, but we are also looking at the prevalence of molecular markers of resistance. And we are also doing ex vivo susceptibility assay, and these two outcomes are actually done by Alfred and his team. And the study is carried out in 3 sites to cover the whole country. So URR, CRR, and then in Brikama at the West Coast region. The second study we carry out this year with the National Malaria Control Program is the surveillance on the SMC drug resistance. So SMC, as you now know, is a combination of SP plus amodiaquine. And since it is given at large scale, especially in upcountry, we need to do surveillance on the markers of resistance. And on the right-hand side, you can see we are looking at DHFR, DHPS, mutation in DHFR, DHPS, PFCRT, and PFMDR1. This study is led by Enyu, who is a postdoctoral research fellow. Interestingly, we are covering even more of the country, 25 health facilities and 3 villages in Basse are involved in this surveillance network. And the interesting thing is that because it is embedded in the GSM study done by Alfred, we also have retrospective data for the last 3 years. So this will give us really comprehensive data on the extent of drug resistance to SP and amodiaquine. Coming soon, meaning early 2023, we are going to launch a phase 1B study on a new malaria vaccine, which combines actually 2 antigens. One R21 is already in phase 3 in Burkina and other countries, and I think Professor Alidou Tinto has communicated very encouraging results in Seattle during the ISTMH. And this is combined to another antigen which is called RH5.2 with a virus-like particle, and they will be given both alone and in combination to estimate the safety and, of course, the immunogenicity in children and in adults. So here you can just see R21 plus RH5.2. So this is the design. I'm not going into the detail, just to show you that there are 9 groups. They will be recruited in a staggered way. We start with R21 because this is the one that is already known, and this will get us going. Then we will move to adults with first in human test. And then moving down to infants 5 to 17 months, with each time you can see the dosage and also the administration. It will be either given at monthly interval, the 3 doses at monthly interval, or we give 2 injections at 1 month interval and the 3rd dose at month 6. Just to say this, study will be done in collaboration with our colleagues from the VIT group, because Beate, Ed, and Sophie are also involved in this study. Okay, sometimes to have some fun, we go to the field with the entomologist and we do some really fun things. And this picture shows you a study that was initiated by Steve Lindsay, which are experimental houses called up and down houses. As you can see, these houses can go up, can be moved up or down up to 2 meters. And it has been shown that if you are higher above the grounds, there are less mosquitoes in your house. They have found that there were 68% less mosquitoes in the elevated house compared to the ground house. They are also doing solar chimneys and ceiling fans to reduce indoor temperature and mosquito house entry. These studies are done by the team of Musa Jawara, who is in the room. And if you want to have more details about these studies, you can talk to him. And then Benoit, who is a postdoc entomologist, who is also interested to decrease malaria transmission. So his first project was trapping mass swarms of mosquitoes for controlling malaria. And he could show that this technique can reduce vector density by 44%. And then there was also a reduced malaria prevalence, but of course that was a pilot study. So next steps will be at community level. He had another project also to try to sterilize wild mosquito by targeting M peroxidase called HPX15 protein with small inhibitors as a malaria vector control tool. This small protein is actually responsible of the fertility of female Anopheles. So more with Benoit if you are interested in this topic. And of course, we never forget to do surveillance on insecticide resistance. So he has also a project on the genomic diversity of insecticide-resistant phenotypes in Anopheles Gambia. These are 2 of our social scientists working on malaria, Fatou Jaite, PhD, and then Omar Cissé, who is starting his master, and they work with Professor Koen Peters. They have done a lot of work trying to understand the perception of communities of malaria interventions, such as mass drug administration, or for example in the ROAST study, where actually households were treating themselves. So we are trying to understand the perception of people and how they can adhere to this kind of intervention. We are also trying to understand how people use bed nets, because it's not only good to have a high coverage, but we also need to understand how people use bed nets to ensure their effectiveness. And then this is, I think, the last slide, just to show the extent of our collaborations in Africa, West Africa, but also thanks to Alfred, we are also part to several capacity building networks that you can see on the screen. And so this gives us a kind of continental collaboration, always optimistic. And yeah, so I would like to thank all my colleagues in the malaria research group, people in the field and people in the lab. We are all trying to work together. I want to thank also study participants, because they continue to strongly support us since 75 years. And without providing names, I would like to thank different generations of malariologists, malaria researchers from abroad, but mainly malaria researchers from the Gambia. So field assistants and nurses, entomologists, technicians who are also doing research, because malaria research starts in the field, of course. So I would like really to thank all these people and different generations since the 50s. This is, of course, young Umberto behind. He's there, yes, and Brian also. And a special tribute to this woman on this picture. I don't know if many of you know Dr. Alice Greenwood. We never talk about her, but she was also instrumental in starting Farafeni Station. She supported her husband in his career. She was also a malariologist, because she started the malaria pregnancy studies in Farafeni, and then she handed over to Clara. So this is the link between Alice and Clara over several decades, and that was my last slide. So this is not specifically malaria, but I just take the opportunity to make some advertising for the upcoming course on epidemiology and medical statistics that will be held at MRC in March 2023. Next year. Thank you very much.
I said 2 questions only. Only 2 questions. Okay, one in the left.
If I understood correctly, the intervention was a combination of ivermectin and an anti-malaria. So did you consider to do like a factorial design, that you can see whether the effect is due to one or the other, or really the combination?
Yes, so you are right. That is one of the limitations of the trial. We could not actually disentangle the effect of each drug, but there is an upcoming trial where I think they will be able to dissociate the effect of ivermectin and ACT. So in our trial, no, it was not possible, because we had only 2 arms, And so, yeah, we couldn't do it.
So did you not consider a factorial design, or were there reasons why you only, by design, you took the combination?
Say again?
Was it by design that you wanted to test the combination, or did you consider a factorial design, but were there logistics reasons?
Yeah, so I was not really involved at the discussions at that time, but maybe Umberto can give you the details about the design.
So we didn't use the vectorial design. So we actually used the determinism piperaquine and ivermectin together, because we thought at that time that in any case ivermectin, mass drug administration of ivermectin would not be used alone. Now there are some people that say that maybe it's going to be used alone. But there is a trial in the Bijagos Island run by Analast, where they are comparing didotermism piperaquine alone against didotermism piperaquine with ivermectin. So that at that trial is going to be able to see the difference between the ACT alone versus ACT plus ivermectin. But in any case, in our trial, we saw an effect on vector density. We didn't see an effect on vector parity, which we wanted to see, but this probably is due to an issue of difficulties of measuring vector parity. So we think that the ivermectin had some impact. Of course, I think the main impact was of the antimalarial drugs. Thank you.
So the last question. In the triple ACT, I wasn't quite clear which are the ACTs you're testing. So, I mean, could you describe that a bit?
Yeah, so the two triple, so we combine AL, which is artemether lumefantrine plus amodiaquine, versus AL alone, plus the placebo, of course, because it's double blind. And then the second ACT is mefloquine artesunate, which is an ACT, combined with piperaquine. And then we compare the triple with the standard ACT, so mefloquine artesunate. And then we look if this is a non-inferiority trial.
If AL, AQ, they select reversely, so is there an advantage in combining AL, AQ if they select?
Yes, because their mechanism of resistance are opposite between the lumefantrine and the amodiaquine. And in the other group, mefloquine and piperaquine, they have opposite resistance mechanism. So if a parasite is, for example, resistant to mefloquine, he will be sensitive to piperaquine, because the mechanisms are going opposite direction. That's how they choose actually the triple, the three drugs. They don't choose them randomly. It's based on the mechanism of resistance that are actually opposite, if you want, because, yeah, as I said, parasites that are resistant to one will be sensitive to the other in the triple combination.
Thank you. Maybe you can continue this discussion after. Interesting. So we are at the end of this. You finish? Stop? Okay, sorry. So we are at the end of this session. So we want to thank all the speakers. Also, thanks all the people who are in this room. Thanks again the organizer and to give us this opportunity to share experiences and also to talk about several topics malaria is one of them, and it is a very nice session. And I want to wish you a very nice end of day. And we are moving now to the closing ceremony. So if you have some announcement before that, or we go to the coffee break before, or we continue.
A couple of announcements before we start. It will be a short closing ceremony. One is that the minister has given an apology because he's still in cabinet. They are still in cabinet meeting, and we cannot wait for longer than this. I think we are on time. We'll be able to meet it by 6:30 because that was the original intended time to end the ceremony. The other announcement is that I understand some of the visitors or international guests would like to check out in the morning and get to MRC with their luggages and do a tour. Some have meetings. I know I have meetings with some. If you are checking out, please let is it Motala? Let the green guys know about it. So not the IT ones. The IT are exhausted now, but the other ones. Let them know so that they can organize this tour with Abdul Sise tomorrow. Abdul Sise is over there. He and Usman Seka will give the tour around at MRC tomorrow. And the reason also why they need to know is because they don't have to go to your hotel to pick you up. You can just be picked up from there straight to the airport. I think those are the two questions I had. I mean, if I remember the other one, I will let you know. I think we've got a long day. So we'll start the closing ceremony with a few statements here. And the first person to really give this statement of closure would be Professor Paul Kay, who is the better person to do this. I mean, most of these projects actually needed money and needed funding. This is the person. He's a professor of immunology, but he's the person who has the basket of money. Or he decides the basket, how much to give you. So Professor Kay is the chair of the Infectious and Immunity Board, where most of us do apply for grants and they make the decisions. So who is a better person to evaluate, the standard of our work here and the standard of the conference, the presentation, which, don't worry, I'm sure he's going to give you plus, and 9 marks over 10.
Thank you very much, Hassan. After an introduction like that, I feel I should say I accept free drinks at the bar. So thank you very much. I mean, wow, what a fantastic 3 days. I mean, we've had such a breadth of exciting work going on and such a mix of history, of forward-looking planning, of really exciting science, and I'll come back to that in a minute. I really wanted to spend just a few minutes, it is late in the day, just to mention 3 things, really. The first is, as Sanjay has just mentioned, I had the pleasure last year of chairing the scientific quinquennial review of the unit. And I just really want to take this opportunity to publicly thank everybody involved with putting together such a fantastic paper pack, for doing all the science in the first instance, for presenting it, if you presented at the QQ meetings that we held, and for making those fantastic videos and PowerPoint presentations that kept us going through the day, the many days. What we couldn't evaluate at that time, and I think that's why it's such a pleasure to be here again in the Gambia, is really the critical thing, I think, about what makes this unit so successful, and that is all of you, and the people and the interactions that go right the way from the gatekeepers as we go into the unit, through all the people that provide us with food and entertainment and refreshments, right the way through the PhDs, the postdocs, the platform managers, all of the staff really, including probably the director as well. And really, I think that's a testament to what is really the power and the strength of this unit, is the cohesiveness of you all as a group of individual scientists, but working together in such a fantastically concerted way to deliver impactful research. So thank you all very much for that. The second thing I wanted to say is a little bit more related to the partnership that's evolved between the London School of Hygiene and Tropical Medicine and the MRC unit in Gambia. I'm an alumnus of the London School, and I started as a postdoc at the London School in 1985. And in those days, there was always an interaction with the Gambia. But it was a very strange interaction with the Gambia that involved a very small number of people that seemed to appear and then disappear, and then maybe 6 months later appear again. And we sort of knew that they went to something called the Gambia, but we had no real idea, if you weren't directly involved in principally malaria research at that time, really what happened in this wonderful setting that you have here. And really not even much of the tradition of what happened here. And I think it's just been so fantastically exciting for me to watch from a distance, and more recently being partially involved in how the two groups, the London School and the MRC unit Gambia, have come together in this fantastic evolving partnership. And I really look forward to seeing over the next few years how that partnership evolves and really delivers an increasing quality of fantastic science. So please, I'd love to see how that does develop. I think that would be fantastic. And then the third thing I want to do before I finish is just to follow on from what was said by the MRC Interim Director at the opening of this meeting, John Erdow. And what John did was to describe very nicely how the MRC really views its portfolio of research in global health and the importance of global health. And I'm sure you're all aware in the UK we've had a pretty rocky road over the last few years in terms of funding global health. But clearly, and as John iterated and expounded on, the two Africa units that we have are central to our vision for how global health research is done in the future, and I think that's a very important message. What John didn't really have a time to go into very much was to expound on a word that we use quite a lot at the MRC, and that is the word excellence. Excellence that underpins everything we try to do at MRC. And I think if John had been able to be with us for the last 3 days, he would have agreed with me and probably all of you that we've been exposed to absolutely excellent science. And that science has been in genetics. We've heard about social determinants of health. We've heard about climate change, and I could go on and on and on. What's hard, again, to really see is you could go, many institutions I go to have excellence in all of those areas, but I really don't very often visit an institution such as the MRC in the Gambia, where all of those elements are brought together in such a cohesive and impactful way, as I said before. And I think that is, again, just a testament to the long-term investments MRC's made, the successful management of the units over the years, the staff attached to the units. And as someone who works on a neglected disease and does their clinical activities across Africa and across the world, without the support of an institution like the MRC Gambia, I can only leave you with 4 words. I am very jealous. Thank you.
Thank you very much, Paul. So I'll call upon Umberto to give his closing remarks.
Well, thank you very much, Paul. This is really nice word, and thanks a lot for your nice word. which you're right, when the subcommittee came to evaluate the unit, so every 5 years we are evaluated, and it's quite frightening, or, well, not really frightening, but quite anxiogenic experience. And the issue at this time was that it was not a face-to-face visit, but it was a virtual meeting. And so that added to the complexity of the thing. But I think we did it well, and I would like to thank the subcommittee, Paul, who was the chair of the committee, but also the MRC UK for confirming the support for the next 5 years. So, in terms of closing remark, you know, again, I'm involved in, I'm immersed into this work the whole day, but still, when I see it again, reproposed, I'm really very proud of it, very proud of what we have been able to do. And so we want to really create an environment that allows the development of scientists, African scientists, and also scientists from other places, but particularly African scientists. So I'm just thinking of projecting where we are going to be in 2047. Of course, I'm not going to be here, but but you know, some people in the audience may be in the position I am here now, or in senior position at the unit. So it has been a great 75th anniversary celebration. I would like to thank Professor Hassan Jay, who has really taken up this challenge of really organizing this meeting, and particularly the team around him, which I think is the best dressed team I ever seen. I don't know if you noticed, but every day they had a different dress of different color, all of them the same color but different. Today is olive green, I think. Can you define this? Olive green, yeah. I think yesterday it was violet, and the day before it was light blue. Yeah, that's right. Very, very elegant. You know, I don't think you know, it's one of the most elegant. These are one of the most elegant people I ever seen. So thank you very much. So this is science with style, research with style. So I also like to thank the friends that attend this meeting, all friends. I mean, Clara Menendez, when I arrived in the Gambia in 1990, she was there with Pedro. They just finished one of the largest BedNet projects ever done that show a decrease of childhood mortality by 60% due to BedNets. So they remain friends. I am not going to mention everybody because otherwise I'm going to probably miss somebody, and I don't want to. We are also results of the history behind us. And I think I would like to thank particularly Professor Tumani Kora, who was the previous director of the MRC unit. Who actually steered the unit at time of quite difficult times. I have to say that as a director, I never get bored because every time there is one problem solved, there is another one coming up. But anyway, so far we've managed to sort of maintain the stability, and I'm really looking forward to the next few years. And also, thanks to all our scientists, young scientists, more senior scientists. I think it's clear they're doing a fantastic job. And I think the unit has a huge potential for doing even more. and to really be an institution for change, you know, to make the difference. So I wanted to say something else, but anyway, I mean, I'm very happy. I'm very happy how the celebration went, and thank you all for your attention, patience. We had some issue with the acoustic, but anyway, that shows that bigger is not always better. But anyway, thank you all, and thanks for being here, and for those who are traveling, have a nice trip back. And yes, I want to also say another thing. I think for us, I want to stress again the importance of collaborating with the Gambian government, the Ministry of Health, the Ministry of Higher Education, Research, Science and Technology. And so I really looking forward to continue this fruitful collaboration with them. So thanks again, and for those traveling, have a nice, safe trip home. Thank you.
Thank you, Umberto. Thanks for those nice words and encouragement. With your last statement of thanking collaborators and those also our friends, I think we will recognize somebody who has been with us from the day one, and that is Dr. Kabir Cham, who has been a friend of the unit. He worked with Brian long time ago, also and you've seen it in the history video, as a scientist then, a field scientist. And he's now currently the chair of our Gambia government MRC Joint Ethics Committee. He's the chairperson. And he has been very elated with the whole symposium and encouraged, and has been here with us throughout. Thank you, Kabir. So now I just ask Liam, our boss, our chief, and he's the person, you all know him, director of London School, which is now our parent body. And it's befitting for him to really give the last remarks, closing remarks to Liam.
Thank you, Hassan. Thanks, everyone, colleagues and friends. So just to assert the fact that I'm the boss, I'm going to just do a 2-hour speech, if that's okay. So it comes with the authority, I think. So just a chance to say thank you to everyone involved. Really exciting science, impact on human health. And I think most exciting for me, and quite a familiar feeling it's becoming, has been to see the potential, particularly the next generation of African scientists, many of them female, ready to take on the challenges to really improve health and make a better world. And I think it really is a privilege to do that as a job. And wow, do we really need you? The world is facing many challenges. I wrote them down in case I forgot them, but I'm not even going to tell you about them. You know about the challenges the world is facing, and we need you to help to generate the evidence to guide practice and policy, and to build the human expertise and capacity for the next generations to really translate that evidence into improved health. So you couldn't be doing a more important job. I think the unit's done a fantastic job of moving with the times, and that's one of the things that's most notable. Continuing with the main challenges, but embracing new methods, new techniques, new challenges, and new areas such as climate change, data science, genomics, you name it. But also it's the way the unit works. It makes me so proud, whether it's around sustainability or the way it's so embedded into the Gambian society and health system. And if you just look at the contributions in the COVID pandemic, you can see the amazing work, the contributions the unit makes to the Gambia itself. The unit joined the school in 2018, as people know, and we're still finding our way. I think, and one part of that, I think, is embracing the kind of decolonizing agenda, facing up to our past and the wrongs committed. And I have no problem or hesitation in apologizing for any wrongs or harms caused by anyone from the school or associated with a school during the colonial era. No problem at all with apologizing for what are undoubtedly harms done around the world. But I have to confess I don't like the term decolonizing, because while we need to face our past and learn from it, for me it's all about finding a better, positive way forward, much more than sort of taking something away. And from the school perspective, what I can honestly say that my aim is really for us to unite around aiming for the unit to continue to thrive, build on its success as a regional and indeed global centre of excellence, focusing really on ensuring that the amazing potential is fulfilled, and I think that's our real aim. I want to facilitate greater independence and autonomy, while remaining fully supportive and in a genuine partnership. And a genuine partnership for me means one that is full of trust and mutual respect, and where you deliver more than you would working apart. That's what a partnership is all about. So really, I will close a chance to say thank you again to everyone involved in the event, and the wider work of the unit has left me inspired and offered real hope for a better world, a better country and a better region, and I'm enormously proud, and thank you again.
Thank you. What a nice words really for closing this. Yeah. Thank you, Liam. Thank you very much. Very nice and encouraging words to close to, as a closing remark. So without further ado, I think with Umberto's permission, I declare this ceremony closed.
They should be much smaller then. Everything should be.
Great.